Hi everyone, we want to get started so we keep to time. My name is Neil Hall, I'm the director of the
not interrupt. And it's in the it's some sort of joke. So Canada is in the Commonwealth of Nations too. So like we have the Queen on our money. So. And at my university, we also have a supercomputer. It's called Colossus, and it's a vertical building. It's an old particle accelerator. And it's one of the few, I think it's the only one with this design on the planet. So the computers are on several floors. Yeah. So now let's get started with the plan. So my talk would be divided mostly in 5 parts. The first will be the background, then some concepts in parallelism, and then array and metagenomics. After that, I will show how we can compare samples using Surveyor. And then I will show some interactive visualization. So basically, people buy sequencers and computers. and the sequencers have been around for a while now, and recently we have parallel sequencers. So, and we have also supercomputers. So both of them can be bought for an amount of money, but the trend in genomics is, however, that we need more parallel software that scale with these machines. So we need parallel software that scale with parallel sequencers and parallel computers. Otherwise, we're all like kind of stuck in the past where we use like software that don't scale on computers that scale on data that was generated in an high throughput fashion. So I put a lot of like reference in the bottom of the slide if you want to delve in this topic. So to summarize, we have super sequencers, if you will. We have supercomputers. So we need somehow super software or whatever. So during my PhD, I did create some of software that scale. I created Ray, which is a genome assembler, which can also assemble metagenomes, do taxonomic profiling, and more recently, compare samples. This software is built on top of some sort of library, a programming library, which is called Ray Platform. And the last project that I started is Ray Cloud Browser, which is unrelated, aside from the name, from the other one. It's a visualizer for looking into our data. And in this talk, I will be talking mainly about 5 software and or workflows. So Ray Meta for assembling metagenomes. So Ray Meta is not a different software, it's a workflow. And Ray Community for profiling metagenomes, but not using elements, using kmers. And then Ray Surveyor to compare DNA samples. So this is new. I will show you a use case for that, for an outbreak we had in Quebec City. And as I said, the Ray Cloud Browser. So our original idea in 2010 was to mix sequencing technology to alleviate some of the technology-dependent errors. And the heuristics to do the assembly were described in our paper in 2010. The main idea was that if we take let's say the Illumina data alone, we have a peak coverage, but also the minimum coverage on the left has a certain amount of kmers. So kmers, as you may know, are words of DNA. So if we mix those two technologies, we move the peak coverage on the right, and we also lower the minimum coverage, which means that the erroneous kmers get less coverage, so they can be more easily put aside by the software. So the platform project, the goal was to build a platform for distributed genomic computing. And one of the things that we saw first was that thread-based programming it's hard, and also it does not scale across several computers. On the other hand, message passing is quite easy to understand. You have let's say two actors, and you have a message going from one to another. But it's harder to program if you use like the low-level libraries like message passing interface, MPI. So the solution here was to create some sort of framework to abstract all these complex patterns. So the Ray Platform project, it has 2 programming interfaces, the plugin interface and the actor model interface. No, don't worry, I won't go into the details. And it has 3 runtimes, the actor playground, standard mode, and miniranks. So most of the Ray code is with the plugin interface and standard mode, but more recently, the code for comparing several samples, it's using the actor model. So this platform scales. So what does it mean to scale? If you have a given problem and you compute something on this data set and it takes a given amount of time, let's say you double the number of cores, then you want basically the running time to be half of what it was. So the figure here basically shows that. If we increase on panel A, if we increase the number of processes, then the running time decreases. So this is from a figure from an accepted paper. So some words about parallelism. And so the parallel DNA sequencing are parallel, but they are not alone. There are other parallel assays in genomics like multiplex polymerase chain reactions and things like that. And as I said at the beginning of my talk, there is a general need for speed on the software side. So like this paper by Flisek at the European Bioinformatics Institute, I think, the need for speed is just described both I2 and how it somehow solved one of the issues of speed in genomics. So just to remind you what is concurrency. Let's say we have 1 million sequences and we have 10 computers available. What we want to do is basically we want to give 100,000 sequences to each of the computers so that we cut the running time by 10. And the two main ways of doing parallel computing are threads, which are local to a computer, and processes, which are also local to a computer, but you can have many processes on many computers. So this is the only slide about the actor model. It's something I discovered quite recently. It's a programming model to do parallel computing. And I just say that about it. You have actors, which are basically some sort of component. They send messages to each other, but they can also spawn new actors. There's a nice video on Channel 9 from Microsoft about that. The original guy in 1973, Carl Lewitt, is talking about that. So now the next part, I guess, will be more bioinformatics. So the metagenomics idea was proposed first by Jo Endelsman in 1988. She coined the term metagenome to represent the set of genomes in a soil sample. The main idea of metagenomics is that the bacteria in complex community cannot be grown, cultured easily because we don't know what they need. So the trick is to sequence directly their DNA. And so as I said, you can argue that this field was started in 1998. And most of the tools right now are based on, not most, but a lot of them are based on 16S RNA on the ribosome. So basically, if you have 16S RNA, you just map them on known 16S RNA, and then you know more about what's in your sample. And recently, there were more methods used that used instead shotgun sequencing. Like instead of amplifying only the 16S RNA, just look at everything. And with that kind of data, with shotgun DNA sequencing, we can do genome assembly or metagenome assembly. We can do also taxonomic profiling using marker genes or some other sort of approaches. And we can also mine for, let's say, gene ontology terms and things like that. So this slide was just to say that most of the methods use 16S RNA. For example, the training course tomorrow and the day after tomorrow, I think the title is 16S RNA something sequencing and metagenomics. But metagenomics you can do, I guess, more things if you go shotgun. But it's probably more expensive to see more than just the type of the iceberg of your samples using shotgun. So some concept on metagenomics. So the taxonomy is a branch of science that does classification, so put things in known bins so that we know what we're talking about. And basically, you can represent life forms using a taxonomic tree where the leaves are mostly associated with living species. And the National Center for Biotechnology Information has something that they call OTU, which is something we will mostly hear you hear this term in the clinic sometimes. It's an operational taxonomic unit. It's basically a taxon with evidence in gene banks or ABI sequences. And you can profile taxons using kmer. So as I said somewhere in this talk, the kmers are DNA words of length k. So it's k nucleotides one after each other. And given a taxonomic tree and data, so data can be either kmers or reads or something else. These approaches use mostly the last common ancestor algorithm to classify each read or each kmer at the appropriate level in the taxonomic tree. And another definition, and what I tend to say is that when people say colored, it either means tagged with some sort of samples or labeled with a taxon or a genome identifier. So in the literature, there are mainly three approaches that use kmers to profile samples. There is the one from Argonne National Laboratory in their bioinformatics paper. There is our approach, which is based, the code is in array, so, and then there is the newest approach, scalable taxonomic estimation, which basically looks like the fastest of the three. So with array communities, you can do genome abundance, taxon abundance, which has a good correlation with taxon abundance reported by Metaflim. And you can also profile gene ontology. And one of the earliest methods that used a taxonomic tree to compare samples was UniFrac. Those who are not familiar with UniFrac, UniFrac is a way to compute distance between two community samples using the fraction of the fraction of the branches that are unique to one of the two samples. RayMeta is it stands for metagenome assembly with Ray. One of the ideas is that because some people bin their reads or their reads using the GC content and things like that, but sometimes it may not be accurate. Other methods also bin the parts of the graph generated from the input using the sequencing coverage. But this may also not be that much accurate because the coverage varies with the GC content. Because if there is more GC, then the DNA is harder to separate, then there is less coverage. So the Ray trick, and we're not alone here. I think the other approaches use this a little bit. So you just take the graph and compute local seeds in the graph, which are basically local paths. And then when I say graph, don't worry, my last part is about visualizing these graphs so that they are easier to understand. So the first thing we decided to check with this de novo assembly method was if we generate a simulated metagenome with 1 billion reads and 1,000 bacterial genomes, how much of each of these genomes can be assembled properly? So I think it's like 80% of them were assembled at least 90%, which is good because that's a lot of bacterial genomes and that's a lot of reads. And also, some of these genomes are similar to each other, like strains of the same species. So, and probably that the ones that are not assembled at all, they are probably like very low coverage, because these reads were generated using a power law. Speaking of power law, using the latest common ancestor approach, we are also able to estimate the genome abundance within a sample. The black curve on these panels is the expected abundancy, and the blue dots are the estimated abundance. So it's quite accurate, although not perfect. And the other thing we wanted to check is that can we reproduce the anterotypes as reported by recent papers? So the first one of the papers was the one in 2011 by the group of Pierborg. They reported that there were 3 enterotypes in the human gut. And so enterotypes are basically blood types, but for bacteria and inside the gut. Later, on the same year, it was reported that there was 2, not 3 enterotypes, and it was some sort of continuum between the 2. So can we reproduce these enterotypes with a kmer-based approach instead of aligning the reads? And the answer was yes. This is a figure from our paper last year also. The data is from MetaHit, from the Kernel paper. So as the other science paper reported, there is 2 clusters, one with Prevotella and one with Bacteroides genus. This is a slide just to say some quotes about what people say about Ray for assembly. So from the GigaScience paper, this is the paper about the Assemblaton 2. Assemblaton 2 was a competition for assembling genomes. There were 3 genomes. One of them was the snake. For the snake, we ranked first according to several metrics, except one, I guess. Also for bacterial sequencing, there is a recent paper in BMC Genomics that compared several assemblers, and they found out that Ray was one of the most accurate. And on the mailing list recently, this is not from a paper, but it's from someone influential in the field, Bastien Chevreux. He wrote, should I say Bastien Chevreux? He wrote one of the popular assembler called Mira, and he said that in his opinion, Ray has a really low error rate, which makes it a good assembler for validation. So this is perhaps the most interesting part of the talk on the results side. Sometimes we have several samples, and we just want to find the differences. And if we have a reference, people usually link the reads onto the reference to find the differences. So this is an example of a difference in the graph. It's a bubble caused either by variation or by a sequencing error. So so some definition first. The idea is to compare samples without a reference. So the differences can be in terms of variance, like a A in this sample, but it's a T in the other. Or it can be also in terms of kmer content, which encompass the other one, because if you have a SNP, you will have different set of kmers generated. So if you want to compare the kmers content, you don't need the coverage, because you just want to check if the sequence is there or not. However, if you want to do things like RNA sequencing or taxon abundances, then you probably want to compare the coverage depths normalized by the number of reads per sample. And you probably want another control or whatever. So the concept of comparing samples is not new. You have a set of biological samples. For each of them, you have DNA sequencing data. And then our approach, which is called Surveyor, it's available inside the Ray package. It uses the actor model, the thing I said about the actors sending messages to each other, which scales. So these approaches, like there is I put like 3 good paper about the subject. The Fermi paper by Engley. So Fermi is to compare human genomes without a reference. There is the Cortex paper in Nature Genetics to also compare human genomes, and I think they also reported chimp genomes in this paper or something. And then the last paper, by Zamin Iqbal also, it's Cortex, but for microbial populations. So these 3 papers, they compare samples using like single nucleotide polymorphisms. So what we propose with Surveyor is it's to compare samples, but just using DNA content. So like the kmers I said. So existing methods, they use variations to compare the sample. So genomic words is also interesting because sometimes you don't know if these are genetic variations or just, I don't know, other kind of differences. So the use case for this that I'm going to show you is we had the last year in the summer, a Legionella outbreak in Quebec City. One of the questions was, what's the source of contamination? We had 3 suspect towers, cooling towers, and the data was obtained on the Illumina MiSeq. We had a lot of samples, 22 patient samples. So all of these are like purified bacterial samples, except the second, which was metagenomic sample. And we had 2 epidemic strains from the environment. And then a bunch of other samples, including 5 from the other outbreak that occurred in 1996. So as I said, mainly there were 2 questions. The first, was the strain of 2012 similar to the one of 1996? And which cooling tower was the most likely source of contamination? So this is a matrix. Yeah, it's not really readable. I don't know why. So basically, it's like a macro array, but it's not a macro array. So each of the column and line represent a sample, and the cells, like for instance, this one represents the similarity between this sample and this sample. So the box here represent a sample from the 2012 outbreak. So, and this one, the box here is the samples from the 1996 outbreak. So we can see that the old samples are not similar to the new outbreak, but they are similar within each outbreak. So the similarity measure here, as I said, is the kmer content. So it's how much of the kmer are shared. And in machine learning, this is equivalent to the K-spectrum kernel, which I included the reference in below. So we can flip this similarity matrix into a distance matrix using the distance matrix formula. It's basic algebra. And we can see that the patients are really close with each other. And so all of this was generated using DNA sequencing data and no reference. So with this kind of distance data, we can generate a tree. And we see things clustered together as they should, like the old outbreak, the new outbreak, like the old outbreak is here and the new here. But however, on this tree, the tower samples are probably not really well placed because they are too different, because they are metagenome samples. There are too many kmers in them that there are not in the other. And the other question that we wanted to check is which tower was the most likely source of contamination. And so when I did that, I didn't know the answer. The answer was obtained by another method by other people from the public health department or something. And the answer was the same. Here we see the distance between each patient and each tower. And it's consistently the tower number 2 that is the closest to each patient. So this was computed without a reference and without knowing the fact that it was a Legionella outbreak. So this will work for any outbreak, even if we don't know the source of if it's based on DNA, of course, because if we have a new form of life that uses something else, then we're screwed. And now the last part of my talk, it's interactive visualization. I said that the field of metagenomics was started in 1998 by Joanne Desmond. In 2004, she wrote a review about metagenomics, and one of the figures of this great review was this one. And it looks like galaxy or something, but it's not from space, it's from bacterial samples. It's a microbiota colored with nucleic acid probe. So basically you have DNA sequences that are specific to certain types of bacteria. And I guess they are labeled with fluorescent label. And so at this point, this figure was included in the review, I guess, because visualization is important and speaks volume about the science. I guess it's beautiful, and you can say that certain type of bacteria are present in the sample, but you could do that with a PCR or just something like that, but it would be less beautiful. So visualization is important to reach out to the general public. So Cloud Browser is an interactive visualization software that works in a web browser, but that fetches the data from the web. So if you go on this address, for those who have a computer, you can do that right away. I started this project not to visualize data per se, but to debug Ray, to fix some issue that we found that were hard to understand because, debugging a graph that has 300 million vertices is quite hard. In real time, it's even harder. So usually you just write some code that write checkpoints, and then you have to restart the job. And so this changed everything in that regard. So this is what it looks like. I will show you, if time permits, I guess it does, a little demo. So the front end is written in JavaScript and in Canva, which is a way to draw things in a web browser. And the backend on the web server is all in C++. Just like Ray, it's also all in C++. And it's all open source as well, free to use and free to whatever. One of the first things we had to decide was how to compute the layout of the DNA sequences path that we wanted to show. And there was something called the Barnes-Hut algorithm, which was actually published in Nature. I was surprised because it's a really mathematics-heavy paper. And it's an old paper. So basically, you just take all your DNA and you divide it in regions, and it allows you to compute forces to compute a layout. It's just to show the visualization. This layout does not have really any meaning on the biological side. And so this is sometimes we hear about bubbles. So this is what I call a hairy bubble. There was a paper recently by a Japanese researcher about super bubbles. This is not a super bubble, by the way. So super bubbles are bubbles where in which there is bubbles. So you can infer like some sort of evolution paths, like you have a linear sequence. Then if you have a SNP, you have a bubble, like this one. And then if you have sequencing errors on one of the two variants, then you have tips, dead ends like this. And if you're lucky, or unlucky, depending on which side you sit, if your sequencing error is in the middle of the read, then you will get a bubble inside the bubble. So that's not what we see here, but so just wanted to share the super bubble paper. And it's interactive to the software. So you can, let's say you find an interesting region, you can select your units, whatever, and you get the sequence of this selection. I will get more into what we see in the demo. We can zoom out to get a bird's view of the sample. So here it's probably like, I don't know, like 200 or 300 nucleotides, large. And there was a paper by, that's not the good one. Yes, that's a good one. It's in the archive. It's by the group of Titus Brown at Michigan State University. They found out that some of the parts in the Brun graphs have really high connectivity, and they found that they shown that using they computed the connectivity of each vertices, and they found huge number that were really connected. So here we wanted to check if we could visualize them. They call them lumps in the paper. So if we zoom in, we can see that somehow this lump is attached only here, and somehow cutting this will just wash it away. And it's really high coverage. So it's probably an artifact from sequencing, from the sequencing process. Yeah, so this is how many times? Okay, so this is the demo I want to show you. It's for sample SRS011134. It's a gut microbiome from the Human Microbiome Project. So let's get started. So Recloud browser works really well in all browsers, but if you use Linux, which I guess you do, no, yes. Firefox in Linux, the Canva implementation is really bad, so you have to stick back to Firefox Chrome. Genome.ulevel. How do I go full screen on the Mac? Yeah. Okay. So first, we have to select the sample. I have only one here, so the choice is easy. Then we have to select the section. It's a four-step organization of the data. You have the map, the section, then the region, then the location. So regions are contigs, location or position onto the sequences. So here I have only one section. I could have more than one if I wanted to compare, let's say, two assembler or two samples. And then I have to choose a region. So these are contigs generated by Ray for this metagenome sample. It's quite a large sample. It has, the graph has more than 1 billion vertices. And the kmers are of length here, of length 21. And by the way, this is just useless. It's just to make people wonder. So I will select this one. And then I have to choose where I want to start. So this contigs has more than 300,000 nucleotides. So I know a good place. So this rotating thing is pink because it's the pink sequence. So the basic IDs here is, so we have vertices and edges. If I pick them one, I can, oh, something happened. No problem. I guess things happen. I will reload the page. How do I reload the page? This was a physics engine bug, I guess. Maybe if I just open another tab. Okay, that's fine. So I won't be moving too much around with the vertex. Oh. So the thing is, when I go outside of the screen, there's something of, I think it's a bug, but probably. So we have vertices and edges. I will disable the color just to show you. So each circle is actually a kmer, and we just show the last. So this is the kmer, T, 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 A, C, etc. But here we see a C. So the C is the last letter of this sequence. It's just a way to represent the graph. Because otherwise, if we will be showing all the sequence, it will be too much information. The other thing about so this is the Bruin subgraph. So the other thing about that is we have an arrow between this one and this one. You can see here the arrow. I will zoom in on just to be sure. So this one ends with a C, and so the property of the De Bruin graph is that you have an overlap of k minus 1. So if I move here, this one should start with a A. So it's the same sequence. I don't know if you can see it, but this one, the A, this one, I will zoom in. This one starts one letter after the other one. So using this concept of like walking window, we can cover all the sample. And the health bars on the top, these are sequencing coverage depth value. And these are in the red because they have low health or whatever. They are lower than the average for this region. And we can see this if I activate the graph mode. I get the coverage distribution for this sample. So on average, it's about 27. Another bug. And at the bottom, we see the coverage depth for each location on the region. For instance, this is the most interesting part. I can press the play button to play the thing. So here we can see I will zoom out also a little bit. We can see that we are moving on the left. And this is all I will pause just to say something before we reach the interesting region. So from here to here, we have ups and downs, but it's all, it's a region without any forks, so it's linear. And here we see a sudden increase. So this increase is, it's not sudden, it happens over several vertices. So that's too slow. So we will see soon here Yes. Here you see what I said, this is the two vertices which have twice the coverage, because this region is shared between the blue sequence and the orange sequence. And we can see what are these. The blue sequence is the contig 135, and the orange is the contig 50. And we can look at this from a bird view and see that they kind of share contents back and forth. This is probably a highly polymorphic region, but we have to confirm that. I don't know. And if I go back to the graph mode, here you can see that it will increase and also increase after. So when the coverage doubles, it means usually that you have two paths that joins, and when it halves, it means that you have one path that forks. So what we can expect here is that we will have forks and joins. So that's in Recloud Browser you can explore like the structural view of your samples. Of course, that's not really useful on its own. You have to know a little bit what you are looking for. But I guess nobody does that, so maybe it's interesting. So, and as you see here, the coverage will drop, leading us to a dead end. So here it's either due to lack of coverage, or maybe the software did not go the good way, or maybe something else. How many times? Yeah. Okay. So I'll go with my conclusion, and oh. I'm sorry I look retarded with the Mac, but I don't. Okay. Can you put me full screen? Okay, so that's done. So genomics definitely needs more scalable tools, not just for assembly, but for other types of analysis. They need to be more parallel also, and somehow easier to use. Robustness is something important, because when you move a vertex outside of the screen, you don't want to reload. And kmer-based approaches somehow answer all these requirements. So I would like to thank Nick Lohman for inviting me, and Leslie Parson for organizing my arrangement. I also would like to thank my funding source, which is the Canadian Institute of Health Research. And for compute time, I would like to thank Compute Canada and Calcul Quebec for compute time on two supercomputers, Colossus and Mammoth Parallel 2. Finally, I would like to thank my director, Jacques Corbeil, and my co-director, François Laviolette. And I would also like to thank Jean-François Erdely, who came from France this summer to work on Recloud Browser with me. It was quite an exciting summer. And I would like to thank Eleni Gozaridis for comments and suggestions on my talk. So now it's the period of questions, if any. So thank you.
Okay, well, that was amazing. I think if there's any bioinformatics PhD students in the audience wondering if they've done enough work, the answer is you haven't done as much as Seb has. So that's incredibly impressive.
I didn't present my collaborations.
You didn't present everything you did. All right, well, that makes us feel even worse. Okay, well, that was amazing. Okay, we've got, so first off, I just say, if anyone's watching on the YouTube, well, I know lots of people are because I'm getting lots of tweets thanking me and saying this is how all meetings should be done in the future. I'm not sure if I've got the nerves to do that, because live software demonstration, live web streaming is basically the most stressful day of my life so far. But anyway, it's all going okay, and people are enjoying the stream. So we've got a few minutes for questions. Who'd like to pose a question? I'm going to pass the microphone over so that the people on the stream can hear as well.
So congratulations, first of all. I mean, that everything just looks fabulous. How is you talked a lot about parallelization and spreading things across different computers. So Abyss did this a long time ago, I think, or not long time ago, 2 or 3 years ago. How is Ray different? Because obviously you're getting much better results than Abyss. So I know that would maybe take 20 minutes to answer, but if you have a couple of lines about why Ray performs better than Abyss, even though.
One of the things that makes Ray really accurate is that it does not do transformation on the De Bruijn graph. If you take Velvet or Abyss or Euler, they all take the reads, build a graph, like a De Bruijn subgraph. So when people say they build a De Bruijn graph, that's false. They build a De Bruijn subgraph because the De Bruijn graph is too huge. And all these methods, they will simplify the graph. Like if they have 2 vertices, when one was one child and the other was one parent, then they will merge these two vertices into one that has one letter more. So from that point, that's not a De Bruijn graph anymore. So in Ray, we don't do that. We just compute paths in the De Bruijn graph. We never simplify the graph. Because the problem with simplification is if you simplify the graph for, let's say, to build contig number 2, later you might need the same structure to build contig number 3. But since you simplified the graph, you might have lost something. So that's one reason. The other reason I would say that I'm not sure, but Ray really use all the kmers and all the reads. Some methods use only the first kmer of each read, and so that using all the kmers gives you a lot of information about the coverage. And so that answer the question?
Yes, very well. Thank you.
Thank you.
Very impressive work. Do you have any plans to adapt this to work with transcriptomic data?
So for my PhD examination, not my defense because it's not done, but I had some sort of pre-doc or something. I don't know how it's called in the UK. One of the questions was that, and I had included data from Saccharomyces pombe to do a transcriptome de novo assembly. So Saccharomyces pombe does not have a lot, or I think it does not have at all any alternative splicing. So if you don't have an alternative splicing, I can say that it works. I tested it, and it's mostly like one contig, one transcript. If you do, then it's hard, and yeah. So because the features are mainly the same, you have a bunch of data sources that have different coverage depth, and they are mixed together. So it's similar to metagenomes. Yeah. Do you have questions from the YouTube channel?
I've got, should I check the Twitter? I've got a question if the microphone's coming back.
Yeah.
So, in terms of the Legionella outbreak,
yes.
you were comparing metagenomic samples with genomic samples
yes.
from isolates,
yes.
and then you were able to cluster
yes.
water towers with isolates.
Yes.
Can you make that kind of statistically robust in terms of saying how likely it is that the tower is
So
is
as
the
you
source?
might have seen in my slides, I was careful to say the most likely source and not the source, because we can say for sure, because we saw that if I go back to the slide, we see here the integers are basically the distance between each tower and each sample. And these are basically really similar numbers, especially these two columns. The difference is probably due to the SNPs between the strains. So, and how likely is it? Well, we could probably go in the, maybe this paper by shot something. Or this one, when you use kernel methods, you have you have some statistical formula to infer the differences between two. But I think the answer from the health department, it was obtained using restriction length polymorphism assays. So, and they basically look for some sites and they cut with restriction enzymes and if there is a SNP, that won't cut. So, but regarding statistical significance, I don't have anything to add about that.
Okay.
But I guess that will be doable.
Okay. Okay, excellent. All right, well, thank you very much, Seb.
Thank you.
Thank you. Okay, so moving on to our next speaker, we have delighted to have Chris Stewart from the University of Northumbria at Newcastle. And Chris, you've just completed your PhD. Just submitted your PhD. Okay, so and it's going to tell us about necrotizing enterocolitis. He's just gone to get his pointer. Okay. So I'll just say while Chris is getting his pointer, well, it's kind of talking to two audiences here. It's quite confusing, but thank you for coming. Thank you for coming online. And you know, this is an opportunity that's been at the very start of our webcast. I think didn't have any sound, so we have to just re-thank the sponsors, which is the CGR in Liverpool, MRC and BBSRC, for letting us do this. Okay, Chris. So. Here's your presentation. Oh, there's your mic. All right, great. Off you go. Thank you very much.
Excellent, thank you. Okay, hi everyone. Thanks to Nick for the invitation. Thanks to Neil and University of Liverpool for sponsoring and hosting the event. So as Nick said, I've just submitted, due to get filed in January, so fingers crossed for that. And the stuff I'm presenting today is really the focus of the last aspect of the PhD, and that's the metagenomic aspect. I certainly expect nobody here wants to really hear about DGG. And so it's the metagenomic aspect which forms one area of the latest study, which is also encompassing some metabolomics and proteomics. But this is very much still in its preliminary stage. This data is very hot off the press. So I'm still sort of pulling together the pieces of the jigsaw to create the picture. And when I talk about the preterm gut microbiome, as I suspect probably most people are already aware, it's the interplay between the microbes which are present within the gut. For me, it's preterm infants. And we have this sort of equilibrium status whereby the gut microbiome can contribute to health, but equally, if a dysbiosis event or an imbalance in this sort of stable and diverse gut microbiome occurs, we have what we call a dysbiosis event, and then that can lead to a disease status, sort of depicted here. So we know then that the gut microbiota is crucial to health and disease. And what's now increasingly recognized, and more so with the advent of the sort of in-depth sequencing technology, is that this polymicrobial community exists, and away from the sort of cost postulates of one disease, one organism, we now understand that bacteria play fundamental roles and talk to each other, and there's a whole host of factors beyond this sort of simplistic view. But like I say, for me, proteome neonates are very much the focus of my research. These guys are a very unique cohort, so once bought and literally shoved into little sterile worlds, so little incubators. Any environmental exposure to microbes is limited by both hand sanitation of nurse and staff and parents, and limited also by antibiotics and antifungals, which are delivered on day one of life. So as a result of that, we have a very delayed acquisition of what we like to develop with a stable and diverse community. The reason I'm interested in the gut microbiome is because it's conferred in two disease statuses, the first one of which is necrotized enterocolitis, which we abbreviate to NEC. This is the leading cause of death in preterm infants, and it is an acute inflammatory disease process of the intestine. So as you can see here, you have these regions of quite severe necrosis, It's a difficult disease to diagnose clinically. Patients usually require being at the advanced stages before you start to see signs, typically sort of inflammation of the bowel. And obviously patients who then undergo surgery have downstream morbidity associated with this. That can include short bowel syndrome. The disease itself is multifactorial, so we know that being premature and bacterial colonization are probably the two most significant aspects for its development. Being premature, because it doesn't really occur in term infants, maybe it's about 1 in 1,000, and in those infants it's fairly easy to treat. Bacterial colonization, because it cannot occur in utero when the gut is thought to be sterile, and also because it cannot occur in germ-free mouse models. The other disease of interest is late onset sepsis or sepsis, and this occurs in more infants than NEC, but it's easy to diagnose and it's easy to treat. And this is a positive blood culture, as I'm sure many of you are aware, with signs of suggested infection. So that's stuff like hypoxia or fever. And bacteria can translocate into the bloodstream and colonize various sites within the body and cause damage. And again, like in NEC, there is a high susceptibility in these infants to this disease on the back of being premature and all the triad that comes with that, including immaturity of the immune system. So the aims then are fairly straightforward. We want to explore what's there over a period of time, so longitudinally look at a cohort of patients. We can then investigate what effect, if any, the microbiome is having on disease status, both pre and post-disease. And we can explore influences because we have a range of demographic and clinical information for each of these infants. So we can look at factors such as birth mode and gestational age and other things that we can't really control. And then more interestingly, we can look at clinical management. So what we're actually doing to these infants in terms of antibiotics, antifungals, probiotics, and how that's affecting the gut microbiome. If any of you work with clinical samples, I'm sure you're aware that study designs are very difficult to come by, but over the last 3 years, I've really pushed for decent sampling, and it was more or less achieved, and the nurse and staff have responded really well to it. So now we have a cohort of over 300 babies and over 3,000 samples. We still have some samples within there that aren't longitudinal enough to be involved in this study, but I was able to include 42 patients and near enough 750 samples from the cohort. And I like to split this into 2 arms. So we have an extremely preterm arm, which is 23 to 26 weeks, and a very preterm arm, which is 27 to 30 weeks. Within each, we have 7 disease matched to 2 controls. These are matched based on birth weight and gestational age primarily, as these are the biggest risk factors, and then where we can, every other factor that might contribute, such as feeding. Within the disease group, we typically have more NEC cases because they're more vulnerable. Within the extremely preterm group, we typically have more NEC cases because they're just more vulnerable. So when I talk about samples, I'm talking about stool samples. What we did is extract DNA from all those samples and process them on the recently acquired MySeq at the university. And this is the first run we did on it in terms of the metagenomics. We used the Schloss SOP for both the wet lab side of things and the ultimate analysis and bioinformatics, as well as sort of implement and chime a bit. And we achieved, for pretty much all runs, a coverage of over 80% at a Q30. The majority of reads passing that threshold, so we were pretty happy with this. So the next step is really to start asking questions, and one of the things that's receiving increasing attention is this idea of a core microbiome. The cutoff varies according to study, but if you say an 85% cutoff, you're looking at all samples which are present in 85%, all OTUs which are present in 85% of samples. And by doing that, we discovered 5 OTUs, which corresponded to an unclassified Enterobacteriaceae, an Escherichia, a Staphylococcus, an Enterococcus, and a Legionella. This Enterobacteriaceae is particularly important in previous studies that we've done as well as others. It's the most abundant organism in the preterm gut, and it's never really been classified beyond family level. And as I'll come to talk about in a little bit, this Escherichia was also deemed to be quite an important genre in this case, but we would want to take that a little bit further to species. So to do this, then I was able, because a lot of the samples have more or less 100% abundance of these OTUs, so picking those samples, I then retrospectively cultured them, was able to pick the colonies, which we did MALDI-TOF on, and then I also did some full M60-NES, which we then sequenced, confirmed that the original MySeq fragment matched the full M60-NES using clustal W2, identified the sequence, and in both cases, the MALDI-TOF and the molecular data corresponded nicely and told us that the unclassified Enterobacteriaceae was in Klebsiella oxytosia, and Escherichia was indeed an E. coli as we suspected. So just an overview of the sort of data to get a feel for it. Like I say, the data is still very much in its preliminary stages, but this is a nice visualization tool. It's a PLSDA produced in Simca, so that's partially squared discriminatory analysis. And what we see is that control infants tend to cluster on the right-hand side of the graphs, and those with disease tend to be on the left. And we can typically differentiate between the 2 disease states based on the bacterial profiles. And this occurs for both the extremely preterm group and the very preterm group. There's a little bit more we can confer for this, but in the interest of time, I'll move that on. And then what I increasingly see in the literature is this idea of pie charts, of amalgamating all your samples into a pie chart and saying this is representative of that community. And so we can do that, and we can have a look and say this is a control, so of all the control samples amalgamated, produced a pie chart, and then we can look at, for example, 10 days prior to disease, and then at diagnosis, and you can look for changes in community. And this is all well and good if the profiles behind these pie charts, before they've been amalgamated, kind of reflect what we're seeing here. I know in my data it doesn't, I suspect in other people's data it doesn't either. That's kind of an important point, and so I'd say to take pie charts with a pinch of caution because you still see them appearing a lot. If we were to say this control profile, do our control infants reflect this at all? Well, the simple answer is no. You can see that you don't need to understand this, it's kind of just there to prove the point. We have a set of core to use, which are represented in dotted lines, and the solid lines represent the sort of satellite community, which are just below the sub-community in terms of abundance. And you can see that as time goes on, these infants develop very unique, individual, and seemingly random profiles with these satellite communities coming in rather transiently. And therefore, a simple pie chart just isn't going to tell us an awful lot of information. Certainly that's not accurate or true. Like I say, again, this is still sort of preliminary, so the hope is to do some modeling, but to get an idea of what's going on in the diseased babies, if you look at the extremely preterm group, so the most vulnerable babies, we see this E. coli, which as I mentioned before, so now we know it's E. coli, and that's represented by the salmon colour. And it's quite abundant within this group. What we interestingly see is that the red lines, which show the point at which NEC was diagnosed, is that we have quite high levels of Escherichia in all cases. Sometimes it comes in, but it's usually one of the abundant species in the good at the time, again here, with the exception being the bottom profile. Interestingly, this baby was the oldest of this extremely preterm group. The dotted blue lines represent sepsis, and typically what we see is that what was cultured in the blood is we see as being a dominant organism in the gut. Rather interestingly as well is this top profile here, and this second bottom profile here, both cases, well, NEC was diagnosed subsequently here, but these were the only 2 fatalities in the cohort, and the last samples we have from these babies, which is why the sampling sort of cut off quite early at day 28 and 23, is absolutely dominated by this E. coli species. So I only actually obtained the E. coli classification at the start of the week. We're now going to go and do some strain work to hopefully take that a little bit further, and indeed try and work out its toxicity effects. Then if we look in the older group, so what I've classified as the very preterms, because they are still significantly preterm but not as vulnerable as the previous group, we can see that straight away the Escherichia species, or the E. coli species, is a lot less prevalent, with the exception of potentially this patient here, where we see a similar trend in over time, and it generally increases in abundance just prior to diagnosis there. We have a lot of sepsis cases in this group, and again we've confirmed this sort of previous results that what is generally abundant in the gut at the time of diagnosis is usually what's found in the blood at that time. Again, we have an exception to this rule of this sort of E. coli dominance, and this guy had a dominance of Staphylococcus with a Streptococcus just coming in and disappearing prior and post NEC respectively. So although we see trends, it's not always the case, and again that kind of goes off the pie chart point, suggests one thing, but if you don't see that, more often than not, you need to take them results with a pinch of caution. So overall, we can show that the development of the gut microbiome in preterm infants is highly complex and highly individual, and now the next step is definitely to apply some modelling, certainly in terms of you guys this afternoon and later this evening. If you want to come and have a chat with where you might suggest I take this data, I'd be more than happy to sort of have them discussions. I've also shown, so this was the first preterm study where I've actually split according to gestational age instead of sort of grouping them all as one, and by doing that I did sort of identify particular things such as the E. coli being more prevalent in the preterm group, as well as other factors which I haven't shown here, but it was found to be statistically different just between those 2 gestational age cutoffs. And in the majority of cases, so not all, E. coli was associated with the pathogenesis of the disease, and interestingly, this species appeared more in the most preterm infants. So with that, I would like to thank my prof, the PI of the project, Professor Stephen Cummins, Claire Lang and Darren, who's here today, who helped out with a lot of the sequencing, the clinicians from the hospital who have been absolutely imperative to the success of any of the work we've done, and in the lab, Giles Holt specifically for running the stuff on the MySeq, and to my sponsors, Tiny Lives, the charity based at the local neonatal intensive care unit and Northumbria University. With that, I'll have a shameless Twitter plug, and I'll take any questions. Thank you.
Great.
I didn't realise there was a little timer. I wouldn't.
Thanks, Chris. Thanks for sticking to time. Who's got a question for Chris?
Hi, I just wanted to ask whether there'd been anything done to look at whether these babies are genetically susceptible to necrotizing enterocolitis.
No, not as far as I'm aware, certainly not by us, and I'm not aware of any of that in the literature. It would be interesting to do. We looked at twins. We didn't know any sort of genetic information, but we obviously knew which ones were related. We found, again, that's the stuff that sort of got me here, but I didn't talk about today because it's been published. But what we see within twins is this sort of similar trend where the sort of, if we have a twin that gets neck and a twin that doesn't, we see that in the one that doesn't. We have quite a nice diverse community. In the twin that may then go on to develop a disease, we see a dominance. In the sort of arbitrary case that got published in the PLOS paper, it was Escherichia, which is the same OTU as what I'm presenting here as being quite a sort of dangerous guy. That's not to say it's always the case. It was only one set of twins which were discordant for the disease, but clearly genetics, in the sense that you can get discordant twin pairs, aren't completely imperative, but they may have important influences for sure.
Did you use the...
hang on, so we'll just do it on the microphone.
For your principal component analysis, did you use single value decomposition or again decomposition?
So on the PLSDAs, on the SIMCA,
The one that you on the left was like
Yeah.
LT on the right.
yeah, they were PLSDAs. So whereas PCA is completely unconstrained, we give these grouping. So the software will try and discriminate those samples.
Okay,
So it knows which one's got neck and which one was pre-neck and post-neck. Also, which one was a control sample. If it can find a difference, is the easiest way of explaining it, it will find it. But by the fact it did find it, it suggests that there is differences there. It's just trying to tease them out.
Okay, thank you.
Yeah.
Okay. All right. So in the interest of time, I think we'll stop there. So thank you very much, Chris. That's great.
Just use that.
Oh, leave that for us, please.
Telly. So if you are watching live, then we're at the Beatles and Bioinformatics meeting at the University of Liverpool. And so the next speaker, great pleasure to present Chris Quince from the University of Glasgow, who's going to talk about a project that we've both involved in. And Chris is a mentor of mine on my fellowship. So you kind of had to invite him, basically. So, are you guys ready? Okay, brilliant. Let's get your presentation up. Okay, Chris, over to you.
Thanks, Nick. Well, thanks to Nick for inviting me and Liverpool for organising this. I'm going to talk more about the bioinformatics, a bioinformatics problem again. It's not working. Oh, okay. I'll stand here. All right. Okay. So yeah, I'm going to talk about a bioinformatics problem, an algorithm I've been working on in collaboration with Nick and Anders Andersens and quite a lot of people all over Europe actually. So that's nice. We actually developed this as part of a hackathon this summer where we got a group of people together and coded up this algorithm over a week or so in Lisbon. And so that was a lot of fun. But it's related to Sebastian's talk because it's an algorithm for something that you can do with your metagenome contigs when they've been generated by the assembler. Now, the problem with metagenome assembly is that it's very hard. I would never dream to actually try to attempt to write an assembler like Sebastian did. It's a very difficult computational problem. And you're going to end up generating tens of thousands of contigs. Now, you have a similar problem with short-read data from genome assembly. But when you're performing genome assembly, you end up with a fragmented genome from the short reads. you know that all those contigs have come from the same genome. When we do metagenomics, we don't know that. So one of the key questions that you want to answer for your contigs is which contig came from which genome. And that's the problem that we've tried to address. Now, there are existing algorithms for binning contigs, and they rely on composition. So they rely on the signatures of kmers. So the idea is that different organisms have different kmer signatures. And that's probably because different organisms have different tRNA codon usages. But now that we're generating big metagenome data sets, we have an extra source of information. So typically in a metagenome study now, you might be able to generate 30, 40, even hundreds of samples. And that provides an extra source of information, which is coverage across samples. So if you think about it, if you have a set of contigs that derives from the same genome, then they effectively correlate across samples. Wherever that organism is, all those contigs appear. So we can use this, this correlation information to do a better job of clustering our contigs together. So I'm going to give a brief overview of the algorithm itself, and then we've been testing this with an in silico data set, which comprises 41 different genomes across 64 samples. I'm going to talk about how we can use read pair linkages to improve the results, and then I'm going to apply it to a data set generated by Mark Palin and Nick Lohman from this 2011 sugar toxin E. coli outbreak. Okay, so how does it work? The algorithm's called concoct, basically standing for clustering contigs on coverage and composition. And the first thing we do is a bit of data pre-processing. So your first step would actually to do a big co-assembly. So you'll assemble all your samples together, and then we take the individual reads and map them back onto the contigs. So that for each contigs gives us a coverage profile across samples. Now, then we do a variety of data processing steps. We'll generate a composition profile, and we want to use both the coverage and the composition data together. And we use actually quite a crude method of incorporating those two types of information. We just stick them together into one big vector. Now that vector will now be very high dimensional, about 250 dimensions. To reduce the complexity of the problem, we just do a principal components analysis and keep the number of dimensions that maintain 90% of the information. And that will be more like 30 or 40 dimensions or so. Okay, so I said we've got this mock data set to test the algorithm on. We generated this data set by taking actually 64 16S samples from the HMP project. So there we've profiled the abundances of different gut the abundances of different gut microbiota have been profiled across 64 different individuals. And then for each of those 16S OTUs, we searched in the NCBI for the most similar genome we could find. And if it was within 5%, we included that genome in our study. Obviously, we didn't, if multiple OTUs mapped to the same genome, we use the most abundant OTU to represent that genome. So using this, we can generate a simulated data set with realistic abundance profiles, and then we simulated the generation of metagenome reads from those. So for each sample, generating reads at random in proportional to the abundance of those genomes, and then choosing a read location within the genome at random with realistic error profiles, but also realistic insert profiles taken from an equivalent high-seq run. So this would correspond to the abundance profiles themselves. So across the top here, we've got the 64 different samples. Each row corresponds to a different species. So we've got some species that are highly abundant. Bacteroides vulgatus, for example, is found in most of the samples. And then some intermediate species that are being found, 50% of the individuals. But then also some very rare species as well. So this is a sort of, it's a realistic test data set. And then we did our co-assembly. We actually used the Ray genome assembler with a kmer size of 41. We got 35,000 contigs and then 50 length of about 32 megabases. And this shows a sort of contig size distribution profile. So we had a few very big contigs, about 100,000 base pairs, lots of intermediate sized ones, and then many more short contigs. Okay, so having done the pre-processing that I briefly sketched over, we can then plot all these contigs in the first two PCA dimensions. I'll emphasize that we're going to use all the PCA dimensions for the actual clustering, but we can use the first two just for visualization. And I've colored the contigs by the species that they derive from. And you can see that they clearly are forming clusters. So this suggests that we are going to be able to devise an algorithm that's going to be able to group these contigs together. The question then is what form should that algorithm take? And what we decided to use was a very basic form of mixture model, perhaps the simplest sort of mixture model you can have, which is a mixture of Gaussians. So by a mixture model, we're going to describe the complete data set as the sum of simpler distributions. So we have a separate distribution for each cluster, and that distribution is a Gaussian with a mean and a completely flexible covariance matrix. And then we fit the clustering, fit those Gaussians with an expectation maximization algorithm. And then having done that, we have the responsibility of each cluster for each contig, which is effectively the probability that the contig derives from that cluster. We have the key question of how do we decide how many clusters are present. And once again, we used a fairly standard approach, which is the Bayesian information criterion. So we fitted different numbers of clusters, and we find the cluster number that minimizes this BIC value, which is basically, it's the sum of the model fit plus a penalization term for the number of parameters. And so for our test data set, we predicted 56 clusters, and I've highlighted the clustering there. So it looks pretty good, but obviously we want to quantify that. So we use three separate statistics for quantifying how good the clustering was. The first is the, I've got those the wrong way around actually. The first is the precision, which is basically how pure each cluster is. What percentage of contigs in a cluster derive from the same species? And as I said, I've got those two values the wrong way round. The precision is actually 0.97. So we've got very pure clusters, but the recall, which is the proportion of a species which is in the same cluster, was somewhat lower. So we got 84% for the recall. So what that's suggesting is that the clusters are pure, but some species are split across multiple clusters. And you can summarize these two statistics in something called the adjusted Rand index, which is a measure of how good your clustering is overall. But I won't go into that in detail. Another way you can evaluate a clustering, and this is useful when we come to real data sets, is by looking at what we call single copy core genes. So we went across all the bacterial genomes that we could download off the NCBI, and we looked for genes that are found in all genomes but only once. And we identified 36 of those genomes. So we can test how good our clusters are by basically a perfect cluster would be one that has all these genes in it but only one copy of them. And what you notice is that we do pretty well with a large proportion of the clusters. We've got some here that don't have 36 core genes in, and there'll be those fragmented species that I mentioned. But we also have a couple that have too many core genes in. So these are chimeric clusters. But I would emphasize that there's only 2 of those across the whole data set. We also looked at how many samples you need to deconvolute this number of genomes. So perhaps the key thing to look at here is the adjusted Rand index and how it varies with the number of samples that we use for the mapping. And basically, it doesn't really start to decline until we get to about 16 or so samples. So although we use 64 samples to cluster these contigs together, we could probably have got away with 16. Okay, so as I mentioned, there's an extra source of information, which is paired end linkage, and that occurs because these are Illumina paired end reads where either end of a longer fragment has been sequenced. So you basically know that for those paired end reads, both of those reads should derive from the same contig. Now, we didn't use that for scaffolding. we used it as a heuristic to join these clusters together. So as I said, we've got pure clusters, but some are too fragmented. So we basically said if a large proportion of the links from within one cluster are to another cluster, then it's likely that those two genomes really were from the same species. Having done that, we can substantially improve the recall to over 90% and get a higher adjusted RAND index. And I didn't really explain this before. This is a confusion matrix. So it shows you for each species the abundance distribution across the clusters, or vice versa. So a perfect clustering would correspond to one yellow blob for each cluster and for each species. So the fact that some species are split, you can see that you've got multiple clusters for some of the species. And then there's the two chimeric species, that will be two chimeric clusters, that will be a cluster split across two or more species. Okay, how am I doing for time?
Yeah, you've got a few minutes.
All right, 5 minutes, that's great. Well, okay, so in the last 5 minutes, I'll just summarize the application of this algorithm to a real data set. So this was taken from the STEC outbreak in 2011. This graph just shows the dynamics of the outbreak itself, the number of incidences over time. And around this point, the genome was sequenced. So the question that Nick wanted to ask, in fact, is whether you could use metagenomics rather than genomics, which has a number of advantages because you can avoid the culturing step, in a diagnostic sense on this outbreak. So can we recover the complete outbreak genome from metagenome samples from these individuals without any culturing at all? So there were 53 samples in total. We had 17 million reads per sample, and after a big co-assembly, we ended up with 22,000 contigs. Applying concoct to this, we predicted 31 different clusters. Now, in this case, we don't know exactly which contig came from which species, else we wouldn't be needing to do the clustering. But we can taxonomically classify about 30% of the contigs. And when we do that, we find 51 different species. So it might look like we're underestimating the diversity somewhat, but only, I think, 17 of those species had more than 5 contigs classified to them. So the species assignment is going to be inherently noisy. So probably this represents a fairly good estimation of the species diversities in the samples. We can look at the confusion matrices again, how the different clusters are separated across species, and we can look at these single core gene profiles. So about half the clusters are looking like fairly complete genomes. We've got multiple clusters from some species. Perhaps not unsurprisingly, there's multiple clusters from the E. coli, which as I said, is going to be predominant in a lot of these samples. The clustering looks pretty good. We have 94% precision. So 94% of the samples from contigs from a species, or from a cluster, are from the same species. Okay, but then the key question was, can we identify the outbreak genome? Now, this was a hard problem, actually harder than in Nick's original paper. In Nick's original paper, he had some control samples. So he had healthy people and he had the sick people, and he identified the outbreak contigs by comparison between the two. We actually only had the people that were sick. So that's quite tricky, but we did have some extra sources of information. And that was the number of days since the onset of diarrhea. So basically, how long ago did these people get infected by the E. coli? So I took the clusters and I looked at their abundances across samples, and I correlated that with the days since onset of diarrhea. And 10 different clusters were significantly correlated with days since onset of diarrhea. But there were 2 clusters that were most significant and negatively correlated. So what that's saying is that those clusters are decreasing in abundance over the course of the infection, so they would be very good candidates for the outbreak genome. And in fact, those 2 clusters were assigned to E. coli, So they actually correspond to the red cluster and the orange cluster on that PCA plot. Now, why were there two clusters rather than just one? Well, you'll notice that one of the clusters has a lower variance than the other one. And what we think is that that corresponds to the core genes of the E. coli, whereas the more variable cluster corresponds to the accessory genes, which are more specific to the STEC outbreak. Why the accessory genes have more variability? They may have more variability in kmer composition, but also there may also be an effect of multiple E. coli strains, so that the core genome will have a higher coverage because other strains are mapping to it. And just briefly, and a nice thing you can actually see is if you look at the position of the clusters along this PCA plot, as we move from left to right, we actually move up in that direction on the heat map. So as we move from left to right, we move from negative to positive correlations with the day since onset of diarrhea. So really, the first and predominant PCA dimension is actually extracting out the signal associated with the infection, which is really neat, I think. Okay, and then, so to validate this, we've got the known E. coli outbreak genome, we map the contigs onto it, and yes, indeed, the vast majority of contigs that derive from the known outbreak genome came from our two clusters, D7 and D18, with more of the core contigs coming from D18 and more of the outbreak coming from D7. Okay, so I think I hope Concoct is going to be a useful program for metagenomics and analysis. It turns these thousands of contigs into much easier interpretable units. And once you've got those units, you can think of many things that you might do with them, trying to reconstruct metabolisms. You could even go back and try to redo the original assemblies. We did succeed in getting the outbreak genome. Oh, I should have mentioned, actually, if you add linkage, then it links those two clusters together into the single outbreak genomes. But actually, I didn't really go into this, but we also identify not just the outbreak genomes, but some organisms that might be important commensals, ones that increase during the recovery period from the STEC. And we also have determined some evidence of co-infection by other organisms. Okay, and then, as I said, lots of people worked on this program. Anders and his students in Sweden, my students in Glasgow, Nick and Joshua, and also Mark for the original idea with looking at the STEC outbreak. Right, thank you very much.
Thanks, Chris. That's brilliant. Who's got a question for Chris? We'll take one question, I think, maybe two. Oliver, okay.
Thanks. Great talk, Chris. So my question is more from the practical side of things. Anybody who's ever tried co-assembly of metagenomes knows that this is like the easiest way of killing your server. So I guess my question is, if you do something like digital normalization to reduce memory requirements, will this still work?
Right, that's a very good question, and one we're trying to identify for ourselves at the moment. So our latest study actually comprises 96 gut microbial metagenomes. We wanted to do a big co-assembly of all the samples together. And even with access to 1,000 core server and the distributed abilities of Ray, we don't believe that we're going to be able to do it. So we're going to have to try something like digital normalization. But I want us to try it out on this synthetic metagenome first to see what impact it has. I think the first thing we're going to try is randomly subsampling the reads and just use 10% of the reads rather than all of them, which seems a bit stupid in a sense, right? You sort of, and a bit disappointing. So, yeah. No, we're going to try it out and see how it works. Yes, that's.
Any pressing questions? No? Okay. Can I? All right, thank you very much, Chris. Okay, so our final speaker in this part of the session is Susanna Salter from the Wellcome Trust Sanger Institute. And in yet another first, we're now having a Prezi, so we're going to swap the laptop over and try that, and we'll see if we can get that going on the live stream as well. So. I think we've set the bar quite high at this meeting. Everyone is now saying that all meetings should be webcasted and like this, and I don't know how feasible that's going to be, but it's quite nice that you can actually just stream this on YouTube without having to pay anything, which is kind of quite an amazing feature, really. You have to have 100 subscribers to your channel to do it. Okay, so have you plugged your thing in? There you go. Okay, so for those watching on the stream, after this presentation, we're going to cut for a break, and we're going to have some coffee, and you can have some virtual coffee, I guess, or whatever you do. And then after that, we're going to have another set of talks after a 20-minute break. And our second keynote of the day is going to be Daniel Hewson, which we're extremely excited about. Okay, so are you good? All right, excellent. So I'll get this out of your way now.
Okay. Is it okay if I sit? Is that all right? Oh, I see. Okay, no, that's fine. I have lots of bits of paper and things, you see. Okay. Yeah, no, that's no problem. Okay, so hi, I'm Susanna Salter, and I've come today from Hinxton near Cambridge from the Sanger Institute to talk about contaminants and contaminant DNA in commonly used kits. I hope everyone can see this okay. Contaminant DNA, as everyone can understand, can be introduced really at any point where you are handling your samples or where you're handling the DNA that's been extracted from your samples. However, this isn't actually always the fault of the person who's handling them. It's been well established over the past few decades that PCR kit reagents in particular are often contaminated with bacterial DNA. This is obviously a real problem for PCR reagents, because if you are using a low biomass sample, then the contaminant DNA in the reagents may be actually more abundant than the DNA you're trying to amplify. However, less widely discussed is the potential for there to also be DNA coming in your extraction kits. This is a real issue for low biomass samples of all sorts, because very little DNA may be gotten out of them. And although it's problematic for all types of study, I'm going to really concentrate today on microbiota and both on whole genome metagenomic studies and 16S-based studies. As I've already said, the reason why this is a problem is because the contaminant DNA may, in these cases, be more relatively abundant than the DNA of interest that you're trying to look at, and create false results, which is like an iceberg of apparent diversity that isn't real. Now, I think people are probably wondering why I kind of took this metaphor and ran with it, because it seems a bit silly. It's not actually mine. It's in recognition of this terrifying figure, which appeared in a recent paper, where what they were actually trying to show was that as you go from sort of culture-based methods of looking at what's in your microbial communities down to next-gen sequencing approaches, you start to see all this extra diversity, which is great, except for the fact that, as you will soon see, a great number of the things they describe as being under the surface of their iceberg are actually commonly observed kit contaminants. Right, so what I'm going to talk about today is three main things, really. So firstly, I want to talk about what exactly it is we actually see in these kits, because most people are quite interested in the kind of things they should be looking out for. Secondly, I want to talk about how it is that they can actually create really misleading results if you leave them in your data set. And finally, I want to talk about how you can identify them and how you can remove them from your results to reduce the impact on your projects. So the first thing I wanted to talk about was comparing different extraction kits. The reason for this really is that I don't want to make out that there's a particular manufacturer who's putting out dodgy kits. The problem is really across the board. So this is some data that's been generated recently by Nick and Simon. Essentially what we have here is a pure Salmonella bongrai culture, which was a tenfold dilution, 10 times tenfold dilution series. And the reason why we chose bongrai was because it's something we'd never seen before in our negative controls. It doesn't really pop up all that often. And we also had the reference strain in my lab, and we had the reference genome to hand, so it seemed like a good one to use. So what we see here is basically 200 microliter aliquots from this dilution series were processed with each of the 4 kits. So each of these color blocks is a different kit, and the kits were used on all 10 dilutions, except this one, which is only 4. And yeah, I'm not going to say what these are, because that's not important, really. So basically, this is these samples after the DNA was extracted were whole genome sequenced. And what you can see in these plots is that the dark bars in each column are the reads from the results which map to the reference genome. The lighter colored bars are the reads which did not map to the reference genome, i.e. this was supposed to be a pure culture, so you would expect everything ought to map to the bongrai genome. And we had the genome of the actual strain that we used in the culture. So what you can essentially see is that the more dilute the culture, the greater a proportion of your results are actually made up of bacterial species, which are not the thing that you're interested in. And so if you imagine that this is like a mock low biomass sample, this is obviously quite a big deal. The 4th kit may have actually performed particularly badly because this is only 1 dilution down from a pure culture, an overnight culture. So it possibly didn't actually extract the DNA very well from the Salmonella in the first place. And I've indicated underneath each of these sort of very approximate CFU counts so that you can get an idea of just how much was actually going into these aliquots which were extracted. So sort of 2,000 cells around about this point is where it drops below the 50% reads mapping mark. And over here, I mean, that was like 20 million cells, so that's not very good at all. So what people are interested in is, you know, what's actually in these negative controls. What is it that's coming through from the kits? Now, I don't have the full results from the graphs that I just showed you. However, we did a small experiment where we gathered up a number of runs which we'd sequenced at the Sanger Institute. These were 454 runs. All of them were from human-associated samples, and this is a lot easier to look at when you're dealing with human samples, which, as you will see in a minute. Essentially, these 6 runs were extracted and amplified. These were 16S runs. They were extracted, and the amplification was done in different labs in different countries. So this isn't sort of a local problem, but they were all sequenced at the Sanger. In order to identify these contaminants, essentially what we did was we had tied the samples to the kit batch which was used to extract the DNA. And we had negative controls for every kit batch that was used. So we looked at what was in the negative controls, first easy thing to do. And then we also did a statistical association with whether all the samples that were extracted with a particular kit had particular OTUs only found in those samples. And these are the results that we saw. So I'm not going to go into this in huge detail because I don't really need to. It really gives you an impression just of how many things we see. This is in only 6 runs. Although I should say that quite a lot of these genera were found in multiple runs. First off, we had a variety of actinobacteria. The kind of things in here that we actually see regularly coming up in other runs as well is certainly Brevibacterium and Pterobacter. And also Rhodococcus comes up quite regularly. A lot of these are soil bugs. Obviously, you'll probably recognize that some of these are actually also occasionally cause opportunistic infections or are human associated. But for the most part, these are soil bacteria. We also had fewer, but still several bacteroidetes. All of these are soil bugs apart from Chrysiobacterium, which causes opportunistic infection in neonates. We had lots of chloroplasts coming through, which is something that people usually automatically screen out of the results, of course. We had a variety of firmicutes, and obviously this is bacillus. Bacillus is everywhere. There are human-associated bacilli. There are food-associated and environmental-associated ones as well. Although none of the bacillus OTUs that we saw seem to be the human-associated ones. And we saw lots and lots of proteobacteria, and really this is the important group, so the alpha proteobacteria. Most of these bacteria are soil-associated. Many of them are nitrogen-fixing bugs. These are not things that you're expecting to find in people's stool samples, in people's nasopharyngeal swabs, in people's blood. And particularly important in this group is Bradyrhizobium and Mesorhizobium and Sphingomonas. These things come up time and again. And they really stand out, like I say, these alpha proteobacteria in human samples. If you were looking at soil samples, they maybe wouldn't stand out so well. We also have several beta proteobacteria. And I would highlight here Ralstonia. That's very well established in the literature as commonly contaminating polymerase. For decades, people have been talking about this. And finally, gamma proteobacteria. And some of these are a little problematic because, of course, Acinetobacter, Stenotrophomonas, and Pseudomonas, these are things that you do actually often see in human samples, but we were also seeing them quite a lot in our negative controls. So that's the kind of thing that we're seeing, and many of these are soil bugs, many of them are nitrogen fixing, like I said, which is quite unusual. But this is actually quite an interesting point in itself because in ultra-pure water production, they often use nitrogen instead of air in the storage tanks. And the idea of this is that it will deter bacterial growth in the system. And it could be that the reason why we see so many of these bacterial species which seem to like nitrogen-rich environments might be because actually they're able to survive in these kind of environments. And it may be that in these cases it's coming through from the ultra-pure water. So next I want to show you some real data that's actually been quite profoundly affected by contaminated extraction kits. This is actually one of my projects. And this run had to be repeated. So really I want to show you this because it illustrates the importance of why you need to screen this stuff out and how misleading it can be. So just quickly I'm going to explain that basically this run was a selection of nasopharyngeal swabs from infants in Thailand. It was a longitudinal study, so we had a cohort of children and we swabbed them every month and wanted to look at the development of the infant nasopharynx. And one of the things you might want to look at in this situation is comparing the 1-month samples to the 12-month-old samples, of course, sort of as paired samples. And so that's something you can do here. So this was a selection of 6 infants who all were born in the same month. Therefore, we could tie these samples together as not being seasonally affected. We saw this fantastic pattern. You have time point 1 at 1 month of age and time point 2 at 12 months of age. And this is exactly the kind of thing you'd want to see. You know, we're expecting to see development of the nasopharynx, and that seems to be the case. And even more so, all of the children appear to change in the same way. So you have a clear difference in clustering between the 2 time points. However, if we plot the entire data set from this run, which is what you can see down here, and we were to colour it by extraction kit, which is what I've done, you see that the orange and red extraction kits here and the blue and green extraction kits here sit completely separately on the graph. And this was essentially just bad luck. We had 2 very heavily contaminated extraction kits, and they just so happened to be used to process chronologically the earliest samples. The DNA was extracted in the order that the samples were taken. So the younger samples happen to be processed with these kits. And essentially, all of this patterning that we see is actually an artifact of what was already in the kits. If you take out all of the OTUs, which we identified as being contaminating in this case, what you end up with the whole run is looking like this. And this is what you want to see. You don't want to see any clustering by kits. That means that something's gone a bit strange. So this slide I like to call the microbiota of a kit. Basically, this graph is taking the OTUs which we identified as being contaminants and just plotting those. So what you have on this graph from left to right is every single sample in this run, so it was about 180 samples, I think, and they're in the order that they were, that the DNA was extracted. And so they're divided up into the kits which we use to extract them. And you can see there's quite a big problem with these first two kits here. At the top, these black bars indicate the proportion of reads associated with that sample, which were actually from these contaminant OTUs. So basically, these two kits were really heavily contaminated. One of the things that's quite interesting is that these first two kits, they're both dominated by this light blue color, as you can see here. This is herbaspirillum. And yet, although they both look quite similar, they also have very distinct profiles, very stable profiles as well, as you can see. So, for example, the one on the right has more Pseudomonas, that's this darker purple, and also Ochrobacterium, which is this sort of teal, dark blue color at the bottom here. So, although they're quite similar, they're both heavily dominated by herbaspirillum, they are quite distinct. And also I should point out that over here you can kind of see this mid-blue colour, like around here, that's sort of all around here. That is also in these two, but is much lower abundance, so you can't see it in this graph, and that's Ralstonia. And as I say, that seems to be basically running through the entire run. And so this may be something that is coming in the water and the PCR reagents, because unfortunately for this run, we didn't appreciate the importance of having a negative control for every single batch of PCR reagents. Right, so. So we know we've seen a huge variety of bugs which are contaminating our own runs, and that in some cases they can be tied quite neatly to a specific extraction kit. And they're generally very different to the kind of things you see in human samples, so they're especially rhizosphere-associated things. So they stand out nicely. But what do we see if we go into the literature and we start looking at low biomass studies? So basically, I've selected 8 studies which have been published in the last couple of years. And what I was trying to do was cover a diverse range of habitats. And I haven't said what exactly the studies are because that's not important, really. I'm just using them as an example to make a point. But I think you'll agree they're quite likely to be quite low biomass environments. So we have coral, we have eyes, we have high altitude air samples, frog skin, human brains, contact lenses, tree worm guts, and healthy human blood. So most of these are environments where you wouldn't expect to have a huge variety of bacteria anyway. And what I'm going to do is talk about the taxonomic families, which they describe in these publications as being important or core members of the microbiota. And really, just to make it clear, I want to say I haven't cherry-picked this data. These are things which are highlighted by the authors as being important members of their microbial communities. However, none of these papers actually describe their contaminant reduction methods. None of these papers say what they found in their sequenced negative controls. So this is all quite speculative, but I hope you will humour me. So these are the results that we had. The first column on the left here is what we found in our negative controls. That's not everything we found in our negative controls, but this is just as a kind of a reference point as to what we found overlaps. And as you can see, quite a lot of them overlap. Unfortunately, the colouring hasn't come out very well here. I've highlighted in blue the ones which overlap with ours. So yes, although I've presented this at the family level, actually the genera usually tie up as well between the two, but you'll have to take my word for that. Firstly, several of the taxa which we don't see in our negative controls do actually make sense for the habitats where they're observed. So for example, there are staphs down here in some of these human-associated studies. That makes sense. Also, you got in the, where's it gone? The blood samples here, there's Fusobacterium Legionella. These are blood samples taken shortly after a dental extraction, so finding dental bugs in the blood is actually what they were looking for. So that isn't out of the question. But and secondly, it's important to remember that the environmental bacteria that we see, which look strange in human samples, are not counterintuitive if they are genuinely present in air samples, for example, or on frog skin. That wouldn't ring any alarm bells for you at all. However, the real dominance of soil and marine bacteria in brain samples, for example, and the fact that the blood sample contents didn't overlap at all with the proposed bacterial, the proposed source of the bacteria that was supposed to be in the blood, didn't seem to deter those authors at all. And in fact, in another publication, not one that I've included here, they were looking at skin samples, and they found that they couldn't actually recreate their strange-looking results when they re-extracted with a different kit and used a different sequencing method. And they put this down to methodological differences in the success of the extraction, and that's how they explained the fact that they weren't seeing some of these bugs, which we see as contaminants, in their repeat data. So while we can't say that the majority of the taxa presented in these studies are contaminants, obviously that wouldn't be fair, it is really important to think about why there seem to be these apparently ubiquitous bacteria which were everywhere. So from human samples to space station samples to Lake Vostok, and they seem to be the same things popping up in all of these environments, and that really does need clarifying. So another quite useful thing that people often ask us about is what they can actually do about it. So we've already talked about screening your data and identifying contaminants using your controls, but over the years, a number of papers have described contaminants in PCR reagents, as I said, and also talked about how they can destroy that DNA before the kits are used. So these methods include, for example, using UV or gamma radiation, DNase treatment, or restriction digests. Also, there are intercalating or cross-linking agents that you can use. However, there are shortcomings to each of these methods because in many cases they find that the activity of the polymerase afterwards doesn't seem to be as good. And obviously, if you're using enzymes on your reagents, this is yet more chance for you to introduce even more contaminating DNA into your reagents. So, sorry. And also, I should say that Mike Cox and his student Elena have experimented with filtering their kit reagents, but we haven't yet tested using UV or other methods on extraction kit components. And I should mention that filters have actually been described in the literature on at least 2 occasions that I've seen as bringing in contaminant DNA and creating some really, really misleading results, which in the end were brought back to it actually being contaminated filters. So while it may be good practice to try and clean your reagents, you need to be aware that contamination is everywhere. And you need to be prepared to identify it and screen it out of your data rather than just relying on trying to stop it getting into your data in the first place. So, in conclusion, I'd say it's advisable to consider pre-treating your kit components if you prefer any one of the methods that are described in the literature in order to destroy contaminating DNA before you use them. Whether or not this is possible, you really should take multiple negative controls. So you want to take them for the storage buffer, you want to take them for every extraction kit that you use, because not necessarily every kit will be contaminated with the same things, each PCR kit, and also your ultra-pure water. And not only should you be collecting these controls, because many people do, but then they do a 16S PCR, don't see a band on a gel, and so they say they're clean. You should also sequence them. Because even, I mean, with the nasopharyngeal samples I showed you before, many of those real samples have a barely visible band on a gel, unless you're really, really bumping up your cycles. So it's important to sequence your negative controls as well. The problem isn't really with the contamination itself. As I say, I mean, DNA gets everywhere. And if you're proactive, you can identify it, you can screen it out. The problem really is the logic gap which people fall into where they really accept their results no matter how strange they might look. And there have been a few examples of that recently. So I think probably a really important point that I say last is to maintain a healthy suspicion of all of your results, especially if you think you can detect any patterning that works by sample processing, so for example by kit batch. And perhaps consider it as a warning light if you start seeing root nodule bacteria contaminating your brain samples. So with that, I would like to acknowledge all the people who have been involved in this. So my colleague Alan Walker at the Sanger, who's extremely passionate about this subject, Nick Lohman, who invited me to come and speak about this, but is also involved in this project, and Mike Cox and the people who provided the test data that I showed you, which is Paul and Claudia Turner, who were previously in Thailand. And also the people who actually worked on these samples in the labs, that's Simon, Elena, and Paul Scott. And also my boss, Julian Parkhill, who was kind enough to let me come and speak to you today about the horrors of dirty kits. And with that, I will take any questions that you may have.
Excellent. Scary stuff before coffee. Oh, Linda's got a question. Okay.
Hi.
It's a really interesting talk, especially for people that work in the lab. And, well, have you done any evaluation on, let's say, manual extraction, like old-fashioned way?
Like phenol extractions, for
Yeah.
example. We haven't compared phenol with kits. No, we haven't compared phenol with kits. So that's a good point. That would be an interesting thing to look at. Although also that still involves the use of ultra-pure water often, which itself does seem to be a source of some contamination. So
And I have
there's still
another-
the potential.
Yeah. Well, you know, I guess contamination is everywhere, so it will just sort of give you another indication that it's not coming from other sources.
That's true.
And another one is about your negative control. Is there a mock kind of extraction using just all the reagents through the filter?
Yeah, that's basically
Okay.
it. So when we say negative control from a kit, yeah, it's passing through water through the kit, basically.
I'm just amazed that you cannot see in your negative control a PCR.
In the band, yes,
Yeah.
you do actually. When they're so those two kits which are very heavily contaminated, you did see that. The problem is you've already done all your extractions by the time you do the PCR to see whether the kit is
but it will
contaminated
not
or not.
go on the sequencer list. You
Yes,
know, you
so
will
in
just
that
throw
case-
everything away. I know there's
In that case, because we'd already done it and we had a feeling we were going to need to repeat the run anyway, we asked for repeat aliquots of all the samples. We repeated all the extractions, but we stuck it on the sequencer anyway. And this is what came out. And actually, I'm quite glad that we did sequence it because it was very interesting to see the stuff that came out of it.
Thank you very much.
Okay, one last question.
Wonderful. Just out of interest, have you got any plans to test the library preparation kits themselves, especially with the PCR-free kits that are coming out?
That's an excellent question. Whether we're going to test any of the library making kits, such as the PCR-free kits.
Yeah, that's a good idea.
Yeah, it would be a good idea.
We haven't done that.
Mm.
Okay, I'm not in this, but I mean the next era kit is particularly interesting because it's you use such a small starting amount of DNA, 0.2 nanograms per microliter, and that's where you start to see this kind of problem if you're doing whole genome shotgun type of metagenomics, and so having a negative control on that would probably be a good idea as well. And I think probably the other issue is thinking about sequencing carryover.
We don't even want to get involved with
those
that.
kind of issues in terms of amplicons that are being sequenced between MySeq runs, for example.
That's a
something
nightmare for
also
another
to
day.
think about.
Yeah.
There are a lot of opportunities to get tripped up, I think, particularly with low abundance type of work. Okay, well, thanks again, Susanna. So we're going to take a 20-minute break now, and I just have been reminded to say that if you brought if you had your coat over in the hub and left it there, it may have been brought over. In case you're wondering where it is, ask Christiana. Coffee for 20 minutes. We're going to pause the live broadcast. Be back with 5 more talks in 20 minutes, and then the last thing is. If you did want to come for dinner, I do have a few places left for dinner, only a few though. Paid, you didn't pre-register, come and find me and give me your name and we may be able to fit you in. We're going to go for dinner straight after the meeting and then after the meeting we're going to go to the Cavern Club and go and see Matthew Street in Liverpool. Okay, so let's just thank all the speakers in the first session. Really interesting stuff. Okay. Are we? Okay. All right. Thanks. Welcome back. So we'll get straight into the second session, and very, very excited to present Daniel Husen from the University of Tübingen. Daniel's responsible for some absolutely keystone programs in metagenomics and phylogenetics analysis, including the very widely used Megan software, which I'm sure many of you have used. So I'm really excited to hear about his recent successes, which I think is going to tell you more about. Okay, so thank you very much, Daniel.
Thank you very much, Nick, for inviting me. So Nick asked me to give two talks rolled into one. So first of all, I'm going to talk about a recent challenge in metagenomic softwares that we've been involved in. And then in the second half of the talk, I'll be talking about recent developments or recent new features of Megan, if any of you are familiar with the software. And if you're not familiar with it, you'll get an introduction to it. So at the beginning of the year, the Defense Threat Reduction Agency in the US put up a $1 million challenge to develop software to identify pathogens in metagenomic data. Okay, so there's this website, the Innocentive website, and you can have a number of different open challenges. We only ever had one. Okay, so I don't know who actually has more than one open challenge at any given time. In any case, so this was a challenge. So there's lots of different challenges on this website. There are not many where the award is 1 million. It's usually a couple of thousand, 10,000, 100,000 if you're lucky. So I want to first of all talk about this challenge. Okay, so this is a challenge. So this website is where people who are looking for a solution to a problem put up the problem, put up a prize, and then people who are interested in winning the prize develop solutions. So in this particular case, the challenge was to develop software that can do this. Rapidly analyze metagenomic data, be it DNA or RNA, and usually from a human source, blood sample or something. It has to be able to deal with all kinds of sequencing technologies, and the goal is to identify the organisms present and characterize all non-host organisms, say what bacteria you see down to the level of strain. Okay, and what's this for? This is for identifying pathogens in a complex mixture of clinical samples. And in particular, I guess the Defense Threat Reduction Agency, its main goal is one of its goals is to fight bioterrorism, I guess. So, people start falling down somewhere and you want to figure out very quickly what's causing this, what's the agent. So on the website, they say, what's hard about this? Well, first of all, one of the hard aspects, as we probably can all appreciate, is there's different types of sequencing technology. So you've got to take into account that you might have a big data set, high quality, generated by Illumina, or you might have a much longer reads, but less high quality, and a smaller data set produced by PacBio. And so that was one of the problems. So you need a method that can deal with all kinds of data. And they really wanted something that's really fast, much faster than what we're used to. They wanted, so we'll get back to that later. And so basically, they wanted something that's very accurate, highly accurate, very flexible with regard to sequencing technology, and very fast, much faster than what we're used to. Okay, so this was a challenge, and it was run in two phases. So for the first 5 months initially, there was a competition phase where people could sign up and start working on their solution. There was 9 data sets available. You could download the data sets, analyze them, and then upload your solution to the website, and the website would immediately score it for you and say how well you're doing. And so the idea in the competition phase was to take these 9 data sets and score them and analyze them and get a score of at least 90%. If you can do all 9, then you'd qualify for the second part of the challenge. Second part of the challenge was called the evaluation phase, where then you were to upload your code, and then people would look at that, confirm the performance that and then compare different solutions on the basis of speed, which one's the fastest, basically. So you have to be accurate to get into the final round, but then you've got to have the fastest solution. Okay, so this is what a typical report looked like. So you analyze a data set, upload it onto the website, and then you get a report like this saying, okay, this data set, you've got 92.9 points, and the score is based on 3 aspects. The organism, did you identify all the organisms that are present in the data set? Did you make a correct assignment of reads to organisms, and did you identify which ones are host reads? And then did you get all the genes? Did you identify the correct genes? Okay, so here, this was a successful run. You can see 92 points, that's more than 90, so that was a good run. And so here are some specs of the 9 data sets. So the challenge data sets are a couple of PacBio. You can see how many reads, roughly 100,000 reads, roughly 2,000 base pairs long, a couple of iron torrent, a couple of 454, and a couple of Illumina. These were all human-associated samples, so up to 97% of the reads were just human, but some had far less human. Okay, so what's the approach? Obviously, the first thing you do is you have one of these data sets, you compare it against the human genome to figure out which ones are the contaminants. Okay, that's no surprise. Then you take the rest of the reads and compare them against a reference database of microbes. Okay, so that's... And we spent some time looking into assembly, so there's always a question, do you need to assemble reads to be able to bin them more accurately? Turned out that it doesn't make any difference. You can do just as accurate binning without assembling stuff. So in the end of the day, we left out this step. Okay, so you compare against the microbial database, and for each of these reads, you've got to assign them to some taxon, and then you've got to determine the genes. So there's nothing magical that we came up with, at least, in our entry to this challenge. We just did what most people would do, the straightforward approach. Basically, it comes down to the details. How do you do this in detail? How do you do your sequence comparison? How do you actually assign the reads? How do you figure out what the genes are, and so on? Okay, so there was those 9 data sets, and I guess to really qualify for the challenge, you'd have to be able to do each one of those within 1 hour. It turned out that our solution does them all in 1 hour on a single computer. And so we needed, in any case, you need an alignment tool that's fast and sensitive. There's lots of read mappers like Bowtie2 and so on, but they're read mappers mapping to something that has a very high similarity. Here you need something that is also very sensitive. If you're trying to map PacBio reads against a reference genome, you've got to allow indels and so on, quite a few, otherwise you're not going to be able to map. Okay, so Illumina and 454 reads, you want something that's fast. It doesn't have to be so sensitive. It has to be quite fast because there's more of those sequences. And for PacBio and iron torrent reads, you don't have to be so fast because the data sets are smaller, but you've got to be more sensitive. So you need an aligner that where you can tune the sensitivity, trade off sensitivity versus speed. Okay, and so we have come up with something called SAS, stands for Sequence Alignment Using Spaced Seeds. Okay, and so we developed this for the target platform, which is 64 cores, 512 gig memory, so a standard, relatively cheap computer. But as we heard earlier this morning, being using multiple cores or having a multi-threaded software obviously is important. If you think back to it, people still like to use BLAST, but BLAST was developed in 1990 when computers had like 4 meg of memory and like one processor. So if you're still using BLAST, that's not the way to go. Okay, so we developed this tool that runs with 64 cores. It can index arbitrarily large databases, and so actually we use it to index the whole of GenBank. Okay, and then it's an all mapper. So when you're doing metagenomic analysis, you want to know for read to which organisms does it map. You don't know just one organism. You need to know all organisms so that you can make a safe call of where that read belongs. Okay, and so we're working on a paper that we're going to submit very soon. Okay, so the sequence comparison. First, we take the reads, compare them against the human genome reference, and then the reads that don't match the human, we mapped against the GenBank. But this doesn't work well enough. There's lots of reads that come from the human that will still make it through. You won't see them here, they'll make it through to here, and then you'll assign them incorrectly to viruses, especially if you're trying to call viruses. There's a lot of human genome sequences that map nicely to viruses. So what we had to do, in addition just mapping sequence against the human genome, we also took the human genome and compared it against the microbial GenBank and masked everything in microbial GenBank that looks human, using a more sensitive setting of our aligner program. So here you're doing something that's not so sensitive. Some of the reads are going to get through, but they're not going to be assigned to anything because you've masked everything that looks remotely human. That gets rid of a lot of false positives. Okay, so SAS-N is actually based on a program that we were developing before we heard of the challenge called SAS-X, which none of you have heard of either, because it hasn't seen the light of day yet, but it will hopefully soon. So this was work that we've been doing for metagenomics. You want to take a whole high-seq run of sequences and compare them against a protein database. Okay? Now, if you do that with BLAST-X, it's going to take you on a single computer roughly 29 years. So that's not feasible. There's something called RapSearch2, which is one of the results of the Human Microbiome Project. And we actually run RapSearch2 on a whole high-seq, and it took us 7 months on a single multi-core computer. And now SAS-X, it takes 23 hours. So you can compare a whole high-seq against a protein reference database in 23 hours on a single computer. But you don't. One of the things we learned is you don't actually want to do that because you get 10 terabytes of output. So the old model where you first blast and then you analyze the output to do something further doesn't make sense anymore. So we actually work on a new approach where while you're comparing the sequences, you actually do the analysis, and what comes out at the end is a profile saying these are the bacteria that you have, these are the genes that you have, and so on. And then you can go back. Hopefully, if we ever get the software to do this, you can go back to individual organisms and say, okay, now show me the alignments for this particular organism, for this particular gene. You're never going to look at 10 terabytes of output. Okay, so that's more stuff that's in preparation. Okay, so in the challenge, one of the problems is to assign reads to taxa. We already heard about the LCA algorithm this morning. The basic idea is you have multiple matches against multiple organisms. And say you have a read that matches these three organisms, where do you place it? In the simple, naive LCA algorithm, you place it on the lowest common ancestor in the NCBI taxonomy of that node. Okay, so it turns out that this isn't good enough if you want to win the challenge. If you want to win $1 million, you're not going to get it doing this. So what we had to do, because it's very conservative, as soon as you've got matched something remotely, something outlandish, you get pumped up to a very high node. And so we spent a lot of time looking at different ways to improve this, and the way that seems to work is a two-phase algorithm. First, you run the simple LCA, the naive LCA, on all your reads, and you see which reference sequences, for which reads you only match one reference sequence. And whenever that happens, you increment the weight of that particular reference sequence. So all the reference sequences get weights, basically how many times do they have a unique matching read. And then you do the LCA again using those weights and assign a read to something that covers 80% of the total weight that it covers. Then you get much more specific assignment. Gene calling. So, this is the basic idea. You've got a sequence from GenBank. It's annotated with genes. You've got some reads. You align them against the GenBank sequence, and you want to know, okay, which gene am I going to call? The problem is that usually the same read is going to match multiple genes and multiple reference sequences. And if you just report them all, at least in the challenge, you don't get a good score. And probably in reality, that doesn't make sense either. And so here the thing to do is to take those weights into account that I just mentioned. Something that attracts a lot of reads is going to be more reliable. And so if you hit a gene that's on something that's attracted a lot of reads, that's a better call to make. And also you look at the genes and how much of the gene is being covered by reads. If you're the only read matching that gene, and all the other genes have lots of reads matching them, then it's probably a false positive match. Okay, so we built this pipeline here. So here's your input data. And then here's the SAS program that compares against the human genome. Then there's a program that just, says those guys that match the human genome, we just count those and report them to the output. The other guys get compared against GenBank, depending what kind of sequencing technology. That's a fast and not so sensitive comparison, or a very fast, or a slow and more sensitive. Yeah, and then we do the analyzer at the end, does the LCA algorithm, the weighted one, and then does the gene calling, and produces an XML report. Okay, so as I said, for those of you who've dealt with 6 million reads, say, do you normally, how long does it normally take you to get from the beginning to the end of analysis? Probably more than 1 hour. So we allowed 1 hour here. For Illumina, you're allowed 3 hours. Let's say we can do all 9 in about 1 hour. So that's pretty fast. And so this is how the challenge played out. So it ran initially from January to the end of May. Okay, then by the end of May, nobody had actually qualified for the second phase of the challenge. We heard of the challenge the day before it was initially going to expire. And so we sent some emails around and said, oh God, I wish we'd have heard of this earlier. We could have competed and so on. Well, we took a look at it in any case, and we uploaded our first solution on the 21st of June and went straight into rank 11. You could always see how well you were doing compared to everybody else. So when we saw 11, we said, okay, this looks pretty hopeful. So we dropped everything else, there's 3 of us, for the next 6 weeks, or the remaining weeks, and just worked on this day and night. And then on the 11th of July, we were the first team to actually solve all of those 9 problems. Until the very last day of the challenge, we were the only team to solve all 9 problems. In the last hours, 2 other teams actually also managed to do it. And so in the evaluation phase, there was only 3 teams. And it took from the middle of July to the middle of September. And every now and then I got an email saying, can you modify this? Can you modify that? And I thought, well, they're asking us to modify all these things. It's probably not looking so bad. Yeah, so this is the scoreboard here. And you can see there, we were the first to get the 810 points. Then at the very last day, there was 3 groups that got 310 points. And then a couple of days, I don't know, end of September, I got an email saying, you're a possible winner of the challenge. And I'll be able to do all the paperwork and blah, blah, blah. And then finally, we actually won the challenge. And this was big news in Singapore. That's where Jay Towers pays. So there's 3 of us. There's my PhD, my diploma student, Benjamin, Jay Towers, a postdoc in Singapore, and myself, who worked on this. Yeah. And so from the day that we heard the challenge to the day that the money was in the bank, it was less than 4 months. So it was a pretty good deal. Yeah, so what did we, okay, so, yeah, so what can I say about, what did we learn from this challenge? So for me, it was a new insight that you can actually do DNA alignment and do good metagenomics. So I'd always been a big believer in doing protein alignments because you can use homology to see things that are more distantly related, because in the past, one of the problems with metagenomics has always been that there's not enough reference sequences. But actually here, if you've got a DNA aligner that's sensitive enough, and you're not using a mapper, which is not really made for DNA alignment, but for mapping, then you can actually do can do metagenomics. And so we've developed some new tools for doing high throughput metagenome analysis, and I'm sure that other people competing in the challenge have also come up with much faster ways of doing things. So I would assume there's going to be quite a few papers in the near future where people just describe what they've developed. Yeah, and so yeah, with the tools. So we've got, for example, we've got 90 data sets of each about 10 million reads from a medical study, and now we can just analyze them. It's not a problem anymore. It used to be like, God, if you've got 90 data sets, each 10 million reads, you're going to need fantastic computational resources. The only problem that we have is that we, if you get terabytes of blast output, that's not very good. Okay, so that was the first part of the talk. That was just about the challenge. Yep. And so, now the next part of the talk, I was just going to briefly say some stuff about Megan, which is the thing that I've been working on the most since 2005, I guess. That's when I got working in metagenomics. And Megan's a tool that helps you to solve the 3 basic computational questions that at the beginning of any metagenome project is usually, what are the organisms present in the data set, what genes are present, and you always want to compare stuff. I mean, most metagenome projects, you end up comparing different data sets and correlating them to different external parameters. And all this is computational stuff. That's what I focus on. But obviously, at the end of the day, this only makes sense if you've actually got some real biological or medical problem that you're trying to solve. In the early days of metagenomics, you could do a pie chart saying, this is what I found, and then you'd get a paper for that. That doesn't work anymore. I mean, and it doesn't make sense. Metagenomics, is becoming a tool to actually answer very specific problems and not something in its own right. So who is out there? So the Megan program can be used to solve the who is out there problem. So you've got your sample, you do sequencing, and then you do a comparison against reference databases, get a big blast file at the end of the day, and then Megan will parse that file and produce a taxonomic profile for you. It will bin your reads onto different taxa. So in the past, that's been BlastX, and so hopefully soon it'll be our tool, maybe, if people want to do it that way, or maybe someday you won't have to do any of the blasting. You just go straight from the sample to the result without producing big files in between. Okay, and then you do the Megan analysis, and so you've got your sample, you get your DNA reads, you do a blast, and you get these blast matches, and they give you hints of where the read might be coming from, because most sequences in a reference database like NR are annotated by the organisms from which the sequences were extracted. So that gives you some hints of what the sequence might be, where it might be coming from. And I already said you get multiple hits. I could probably take that slide out. And then you analyze them using the LCA. One way to think of the LCA is also placement by shared gene content. You say, here's my read, I map it to this gene appears in these 5 organisms, they all have it in common, so I'm saying it's one of those type of organisms. And if they're all alpha proteobacteria, then the read gets placed on the alpha proteobacteria node. So everything gets binned onto the NCBI taxonomy. It's now over 1 million nodes. When I first looked at it, it was like 200, well, I think the very first time I looked at it was 235,000 nodes. Now it's 1 million nodes, so that tree is getting bigger and bigger. And so it's a tree, basically, any sequence, any organism that's represented by a sequence appears in the NCBI taxonomy. And it's ordered by, it's structured on different ranks, kingdom, phylum, class, and so on. And so if you open up a blast file in Megan, you see a picture something like this. You see the first part of the top part of the NCBI taxonomy. Here's the root of the taxonomy, cellular organisms node here, bacteria, and then the different phyla, and below the phyla, the different classes, and so on. And Megan just scales the nodes so that you have some idea of how many reads have been assigned to that particular node. And then you can collapse and unccollapse parts of stuff. If you want to see more detail for a particular node, you can unccollapse it and see what's below it. You can produce pie charts if you want to. That's not the most exciting thing. You can drill down to the actual individual reads and alignments, and you can inspect them in detail. Sometimes you want to do that, although it's becoming less and less interesting as data sets get bigger and bigger. But although, still you might be an expert on one particular organism, you want to go down and see the actual reads. So you want to capture the reads. Megan allows you to save all the reads for a particular organism to a file, and it also produces alignments of the reads against the reference sequences for you, so you can go in there and look at those. So the function analysis, the way that that's done in Megan is that reads are sorted into different classification schemes. So it's the same computation. For example, seed. Seed is one way to classify gene function. In seed, there are so-called subsystems and functional roles. Reads are mapped onto genes. Genes play functional roles. Functional roles come together to implement subsystems. That's the basic idea. It's a bit like KEGG, where there's enzymes and there's pathways. It's the basis of the RAS and the MGRAS annotation pipelines. You can think of it as a graph with roughly 13,000 nodes. This is a website there. This is just a picture of a subsystem. Yeah, so Megan draws a seed classification like this. It draws it as a tree. Here's a root node, and then you've got the different type. So the seeds are classified in different there's a number of levels of classification. For example, there's carbohydrates, and carbohydrates, there's different types of carbohydrate-related subsystems, and then they break down into further ones and so on. Okay, so there's a seed, there's KEGG. You know, KEGG is nice because it's got all those pictures of pathways that you can actually look at. So it's a classification of pathways. Again, but you can think of it as a tree. There's these different types of pathways, like metabolism-related ones. There's different types of metabolism-related pathways, like energy metabolism. And one of those is photosynthesis. And if you drill down far enough, we actually get to one of these pictures. And then in Megan, the different enzymes get colored by how many reads have been assigned to them. And you can actually go to one of these enzymes, and you can say, I want to know which genes have been, I want to see the reads and the alignments for that particular enzyme. So KEGG used to be free until 2012, 11. And so Megan still comes with the old free version. If you want to use Megan with the latest version of KEGG, you have to get a KEGG license. But once you've got those files, it's easy to open those files with Megan. So you can use, even though Megan doesn't ship with the latest KEGG, if you have a license, you can actually use it with Megan. Okay, another classification scheme, just the third one implemented in Megan, is COGS. So COGS were around for a long time, and then they stopped generating more COGS, so there's like 3,000 COGS, classes of orthologous groups. And then Per Borg's lab and other people decided to recontinue the classifications. Now they've developed something called Eggnog, and the Eggnog consists of the classic COGS with some new NOGs, which are non-supervised orthologous groups. And so you can use those also to bin your reads. You know, like here, there's cellular process and signaling, for example, and one of them is defense mechanisms, and then you can see all the COGS that get assigned to that. And then again, you can look at the reads that have been assigned to a particular COG, and you can see how they align, for example, to the actual reference sequence. Oh yeah, so alignment. So this is something that we spent some time working on, developing an alignment view in Megan. So here you can see the reads coded as proteins, because this is based on a BLAST-X comparison, but you can see at the very end, you can see the trailing nucleotides that weren't part of the alignment. And in the aligner, you can switch between the protein view, and you can also go back to the initial DNA sequences. And that can be quite interesting. You can see that obviously the reads recruited to this particular gene, obviously they don't come from one genome. You know, and so we've been interested in trying to estimate how many different, I was going to say species, but how many different genomes do you have in your metagenome by looking at alignments to such genes such as RPOB, and then trying to estimate you know, from how many different genomes has this alignment recruited its reads. So we do some kind of sliding window thing, we get some kind of number. I think here the answer is 17. So using our heuristic at least, we get a number of 17 as an estimation of how many different genomes contributed to this particular alignment. Megan allows you to do compare stuff. And the easiest way to do that is just to load multiple data sets into what's called a comparison view, and then do everything that we've seen so far. But you see multiple data sets simultaneously, and you can produce all kinds of plots and so on. This is an arms race that I don't think we can win, because no matter how many plots we implement, there's always stuff that people want to do. So at some stage, I'm just going to give up. And so the plots that Megan can do, it can do. But you know, there's bubble charts, and I get some weird requests for features. This was something that somebody said, you've got to make one of these. Okay, so I put one of these into Megan. I hope that someone out there is using it. That was a lot of work. Here's a KEGG comparison. You can drill down, as we did earlier. And then here, Megan tries to show you how many reads have been assigned to a particular enzyme from the different data sets. But you can see there's a limit to how much you can fit into one of those little boxes. You can do PCOA type analyses in Megan. You can have the different data sets. You know, this is one based on KEGG. There's not enough time to actually explain this in detail, but this is from a permafrost paper. This is the original analysis that the authors did. And then we redid this using Megan, and we got a very similar kind of picture. Yeah, Megan allows you to attach so-called metadata to your samples, and then you can use that metadata to color things, to give them different shapes in pictures, you know, triangles versus circles and so on, to label things in analyses. And you can also do calculations, for example. You can compute the total biome or the rare biome, the core biome, those different types of biomes that people like to calculate. You get a new data set that's based on some summary of the data sets that you've selected beforehand. Okay, look, well, I just want to say that I mainly just program stuff, but I also work with people on real projects so I know what people are actually interested in. One thing that we're looking at, inflammatory bowel disease, and we have germ-free mice that then, are given a specific gut microbiome, and then you look at the interaction between a disease state and microbiome. And there's things that if you can take a mouse that is prone to a colitis, and if by changing its gut microbiome, you can make sure it doesn't get a colitis or vice versa. So that those kind of experiments. You've got lots of data. And there's another thing we're looking at is patients who are heavily overweight and they go through different treatments, either operations or different diets, and we are following those guys, trying to learn about the connection between response to therapy and gut microbiome. Yeah, computational challenges basically, we're getting petabases of sequences. We need exabytes of data. And we heard this morning that we need ever faster analysis methods. And I don't know, how am I doing for time? Yeah, you're okay. Okay, then I get to an important slide. This one here. This is from a paper, 2009, from the GBA project, the Genomic Encyclopedia for Bacteria and Archaea. This is a joint project between the JGI and the Deutsche Sammlung für Mikroben und Zellkulturen. And what they're doing is they're trying to get a better reference database for metagenomics by systematically sequencing bacteria from underrepresented phyla. And so this paper gives a good idea of where we stand. This picture here is really interesting if you haven't seen it before. So here they took 16S sequences from the Green Genes database, 40,000. And if you take those 40,000 sequences and compute what's called the phylogenetic diversity for that data set, you get 1,000, just over 1,000 units, whatever they are. Just a way of measuring how similar those sequences are in a tree, basically. Now, if you take those 16S sequences, for which we know from which organism they come from, you're looking at this here. You're down to under 200 units of phylogenetic diversity. And if you take those 16S sequences for which a whole genome sequence exists, then you're in the blue part here. So you're down to less than 20 units of diversity. And their main point was to show that, yeah, they sequenced some genomes and they made a big jump in the coverage. But what you see in the bigger picture is that if you're hoping to be able to analyze metagenomes based on existing whole genome sequences, then there's going to be a lot of stuff, a lot of diversity that you're not going to be able to see because it's just not present in the reference databases. So, yeah, so, I mean, and that explains, I guess, that's one of the reasons why the Human Microbiome Project has put a lot of effort into sequencing individual bacteria rather than just sequencing human samples, because without the appropriate reference sequences, you're not going to be able to analyze your data. So this is, I'm not sure how far we are now, but we're still probably down here somewhere. I'll be very surprised if we've got that far. Okay, I just want to mention something here. So there's a program on at the Newton Institute in Cambridge, spring of next year. So the goal of this program, so at the Mathematical Newton Institute, is to try to get mathematicians interested in questions concerning metagenomics. So if there's anyone here who's mathematically inclined or would like to try and convince mathematicians that this is a good thing to be working on, it's a 1-month program with a number of workshops built in, so you can either come for a month or you can take part in some of the workshops. So that's something to put in your diary, in your calendar. Okay, and these are all the people that I'd like to thank, and I thank you for your attention.
Okay, excellent. Very good. Well, I'll ask the obvious question. So,
Okay.
what you get, well, firstly, drinks are on you tonight, right? So,
I already bought lots of
what you
champagne.
get? What are you spending your money on?
No, you don't want to know.
Really?
Yeah, it's sad on retirement funds.
That's pretty sad, isn't it? Yeah, well, that's a good way.
Well, I did get a new iPhone.
You got a new iPhone. Okay.
That's about as far as it went.
Okay, we've got a question from the audience. Who's going to volunteer? Well, we haven't got a question from the audience just yet. I've got one from Twitter. Oh, Seb, you go after one from Twitter. So this is how we're doing it. From Surya Sahar. He says, how good is Megan in exporting data out for analysis using external tools like R?
I put a lot of effort into making it easy to import and export stuff. So there's an export menu, and you can export comma separated, tab separated files in many different variants. You can say, I want the taxon name and the read name, or I want the read name and the taxa, and I want the score, and blah, blah, blah. So there's like about 15 different formats that you can, different variations of that. Okay. So I've tried to make it really easy to export stuff, and vice versa, you can import stuff. So you can take different results from different analyses and import them all into Megan, and then see them all together.
Okay, that's good. Yes.
You said that there were about 300,000 nodes in the NCBI taxonomy a few years ago, and now there is like more than 1 million.
Yeah.
So where are these nodes? Are they mostly in the species rank?
There's a lot of garbage nodes. Like somebody did a study on drinking water bacteria, and so you see drinking water bacteria number 1, number 2, number 3, number 4, like a couple of thousand. It's just a whole. I mean, I don't think that's very useful, because the chances that those particular drinking water bacteria are ever going to be rediscovered somewhere else are quite small. So there's a lot of stuff. I guess it's because there's got to be some automatic way that if you assign, when you upload sequences, you've got to assign a name, and that name, if it's not present, it gets put in the NCBI taxonomy. I guess there's got to be. I don't think. I mean, I think a lot of work is being done to make it a good taxonomy, but a lot of those. That increment is just of that nature. Many isolates. There's lots of isolates that put in.
And do you know if there is an electronic version of the Burgess taxonomy?
Yeah, I'm pretty sure there is.
Would it be useful to use the Silva database?
Silva, yeah. So there is actually something called CREST, which some guys in Vienna developed, which is it works together with Megan, and they use the Silva taxonomy. So you can use Megan for 16S, and so some people like to use the Silva taxonomy, and so there is a way to use the Silva taxonomy with Megan. It's not currently, but we're working on it.
And can we, the new aligner, can we use that now, or do we have to wait for you to publish it?
That is a new aligner.
Okay, right.
Okay,
so
it's not published yet.
It's not published yet. But can you send it to us? Because we need it. Okay, you're keeping that close to your chest. All right, fair enough. Okay, all right. Well, thank you very much, Daniel. That was fantastic. Okay, so next we're going to have Sujay Kumar from University of Oxford, continuing the metagenomics theme and telling us about a toolkit that you developed in Edinburgh with Mark Blackster called Blobology. Is that right? Okay, so thanks, Sujay. Have you got your thing on? Okay.
Is that okay? Is it still on? Yeah, okay, cool. Okay, so I'm obviously right in front. Excellent. So this is the hardest act to follow because I've never won $1 million. And Chris Quince did a great talk in the morning, which I absolutely loved because basically it's blobology on steroids is what he's already done. So mine is like the beginner baby version of Concoct. And actually that might be useful to some of you. So I'm going to pitch it at that level where if you're doing a project where you're just, trying to do a genome assembly or something, which is where I came from, then it might be useful to you. We found it extremely useful. And, Concoct is the more sophisticated version. This is the the CBBS version or whatever, I don't know whatever you want to call it. Okay, so I'm currently at the Abu Bakr lab where we do study flatworm regeneration, which I'll talk a bit more about at the end of my talk. And I'm a postdoc there, I've only just started. The work that I'm describing, as Nick said, was done in Mark Blackster's lab in Edinburgh, where basically Mark loves all little wiggly things. So he loves sequencing nematode genomes. So this is where this story starts. What is blobology? Imagine 3 years ago, I'm sitting there at my desk doing what PhD students do at the start of their career, of their PhD, which is not much. And he comes along and he says, here's some HiSeq data from this nematode we're sequencing, can you assemble it? And I'm like, you know, whatever. So it was Illumina HiSeq data, it was 100 base paired end, it was 140 million read pairs. I'd played with Velvet before then, I'd played with Abyss before then, there was no Ray on the scene at that time, I think, I don't remember. But we'd just gotten, I had no big servers, nothing, so we'd just gotten a license to CLC, which is a black box, as you all know, if you've used CLC. You have no idea what it does, and it just spits out results. So I just ran it through there and hoped for the best. And what you got was a bunch of contigs. That's what they look like, you know, and as any good bioinformatician will tell you, the first thing you do is, you look at the file, completely at random, pick a couple of sequences, stick them on NCBI GenBank, and see what results you get. So, sorry, you can't see this very well, but basically, for the first 10 sequences I picked at random because I was in a hurry, I wanted to see what I was getting, you know, have we even got everything right. About 40 hits, where is it? About 40 hits were to bacteria and only 17 hits were to Caenorhabditis. And this is Caenorhabditis. I mean, it's not C. elegans, which is, you know, the first completely sequenced metazoan in the world, but it's still, you know, you want more hits than that. So I was thinking, oh, you know, we have lab contamination issues, what's going on? So to quantify this contamination, I thought, okay, I'm just going to plot everything against everything. That's another classic trick everybody does all the time. You just do XY plots of whatever you can think of. So what was nice was this is GC content on the x-axis down in the first one. So each contig is plotted as a dot. Length is on the y-axis. Doesn't tell you much. There's 2 smears. You could have gotten that from a fast QC plot as well. The second plot is length versus coverage. Again, not that exciting. There seems to be something happening there down there with 2 bits. And then when we're doing GC versus coverage, which were the only things we had measured at that point. By the way, coverage is when you take all the reads and map them back to your whatever assembly you'd gotten, and just to see how deep each, how much of each contig you have, so to say. And we started seeing these slightly clearer things spreading out. And when I took the log plot of the coverage, I'm like, wow, this is really cool, blobs. And of course, the correct term would be to say GC, log coverage, cluster, visualization, analysis. But we just called it blobs and that name stuck, so it's called blobology. So that's what blobology is. Each dot over there is a contig. And when we started, we were like, okay, so these must be different things. Mind you, I didn't know any about the existing literature on this, so that's my naivety showing up over there. We'd basically just take a few contigs from the center of each blob and blast them and say, oh yeah, that's all nematode. Oh yeah, that's all microbacterium. Oh yeah, that's all actino, you know, some other bacteria. I don't actually know much about that. So I was really glad. I'm like, we've identified our contaminants. Now, for this audience, I realized that the nematode is probably the contaminant. You know, most people over here are probably interested in bacterial genomics, but we were interested in the nematode because we were trying to do nematode genomes and study how they evolve and things. So we took that a step further and basically started taking all the contigs, doing a blast against, mega blast against NT, which was not 1 hour, obviously. It took about 3 or 4 hours, but you know, that was very quick for us at the time, and we were like very happy. One thing you can do if you have hundreds of thousands of contigs, which sometimes you do, is pick only a small sample of them, because what will happen is, when you pick a random sampling, you're probably going to annotate the things which are closer to the center of the blobs anyway. So this kind of visual comes up. You can, I wrote a little script that basically walked up the taxonomy like Megan. I actually tried using Megan to do this, but I wanted something I could just pipe through in one go, and I wasn't able to get the command line working as well as I could have, but I would have liked to simplify that. And so, yeah, so you clearly see the big blue blob is Rhabditida. It's the order of which nematodes, of which C. elegans belongs to. And that's a lot of different bacteria down there. I mean, what was nice about this plot was this was the first time we had a visual sense of what was going on in our data. Because until then, you basically see this big abstract FASTQ file sitting on a server somewhere, and you have no idea what's in it, really. So the nice thing with this blob plot was it sort of let you see what's going on, and immediately you can start saying things like, right, I need to make a filter, like a blast filter or something that says if I have a hit above a certain quality to a bacterial database, that's definitely, or if it's more than a certain GC value, or if it's less than a certain coverage value. So we could start devising filters, and that's what we did. So we wrote this up as a tool. We've been using this for 2 or 3 years now, since 2010. But we only recently wrote it up as a tool, not realizing that other people have done this already, which is where it gets embarrassing. So it's not a new idea. In 2009, and this was cool because I think Nick tweeted about this, and then Lex Niederbrag found it on Twitter and said, we've already done this. And I'm like, I'm so sorry, we'll cite you, we'll cite you. So what we basically did was we pitched it as a quick and dirty visualization tool, which you can run as a bunch of scripts on the command line quickly to get these plots. So that's all it is. This is not a new idea. If it's something you guys find useful, please use it. I would say don't use it and use SAS, because that's probably going to be a lot faster and easier to use in the future, but that's the idea. What I am going to show you now, though, sorry, there was one other example while I was doing a literature search. Something like that, which is essentially the same thing. But in both cases, it had been done for bacteria, and it's also, I mean, I think our pipeline is still useful because it says, well, when you have large eukaryotic genomes, how do you optimize, what are better tools, and things like that. So I think we still have some value to offer over here. It's just a visualization tool that I think you should do before you do any sort of big genome assembly project, because it helps you identify what's going on in your data. So where was I? Yeah, so here's one of the examples. This is a Hypsibius dujardini, a tardigrade water bear. They're cute little things. They're very small, and they have these arms that do that. When this was done by Georgios and Mark, I mean, I was in the lab at the time, but they were the ones taking care of this project. So here's a sort of hand-annotated example where they said, okay, now we know what these things are, so this is how you devise a filter. You say, if the GC is greater than 60 and it has a really good blast hit to proteobacteria, which is all those blue contigs over there, then remove those contigs. And then do the same for these others. And then you basically take all the reads that mapped back to the remaining contigs and reassemble them. The advantage is you should get a better assembly, because your assembly algorithms often rely on uniform coverage, and that will get removed because some of the uneven coverage will get removed from removing these contigs and getting a better assembly. We found better assemblies almost always whenever we did this, so that's the good thing. One quick point about this is a tardigrade, but most of the hits seem to be yellow, which is chordata. Sorry, you can't see colors really well, and green, which is arthropoda. That's just because there are very few tardigrade sequences in NCBI-NT. And so what you're seeing is nearest hits, which are invariably to those orders. This coloring can be done at whichever level, species, order, the little mnemonic you'd shown. You can do it at the level of kingdom, superkingdom, whatever you want. Here's a cute little one-liner. This is all, by the way, on our little GitHub where all the scripts are. That's on the next page, the link for it. But how to sort of create using 2 command lines on the Unix command line to get the proteobacteria subset of NT, because you don't want to be blasting against everything if you just want a quick check against just that database. So from now on, the next 4 or 5 slides, I'm going to show you how we use the blob plots to uncover various things about our samples. So this one was for separating endosymbionts. So an endosymbiont was a bacteria that lived inside, a Wolbachia bacteria that lives inside a nematode cell. In this case, Dirofilaria emitis, the dog heartworm. So the first 2 pictures are from a different sample. It was a different strain, but was so close to ours that we assembled them together. The third picture was a different strain, and the fourth one is all libraries combined. So the coverage of all 3 libraries has been mapped back. And what you see is that the red, which is the Rickettsialis order, which is the Wolbachia, is clearly separate. It's by several orders of magnitude, well, 2 orders of magnitude higher over here, but it's sort of in line with the others. So first of all, it tells you which sample had the Wolbachia in large numbers and which ones didn't. What's also nice and not as visible over here is that there are some red dots in the main nematode blob. And those are the examples of lateral gene transfer because they hit Rickettsialis, but they had the coverage of the rest of the genome. So when we wanted to reassemble, in this case, we wanted both. We wanted the Wolbachia genome separately, and we wanted the nematode genome separately. We actually used this as a visual guide, put a cutoff line where we didn't see any red dots. It's a bit heuristic, but then also used the BLAST results to map things back. And when we assembled it, we were able to get the Wolbachia genome back in very, very few contigs, which earlier was in much more, many more contigs. So that's one way of how this has been useful. This is an example where if you remove the contaminants based on a preliminary viewing of this plot, you will estimate the coverage more accurately. Otherwise, you'll end up with a coverage estimate like the assembler. I think Abyss does this, Velvet does this, where they have an expected coverage auto option where they try to use the mean coverage across everything, and then you'd choose the wrong value, basically, and you'd get a bad assembly. Here's an example. This is one of my favorite ones. This was Acanthochelonema vitii, another nematode, parasitic, I think. Yes. And Georges was working on this project, and he comes in, and they're red dots. This is done at the level of, it's saying Chordata nematoda, so it's done at the level of phylum. But initially, we had it done on the level of order, and it came back with primates. And so basically, there's primate DNA there, and we were like, OK, somebody is, bled into the tube. I don't know what happened over there. But we looked at the individual sequences, because we could pull them out, and it was Macaca mulata. So this is the classic quote. I still have it written out on my wall in Edinburgh. I put it, there's a stupid monkey in my worm, is what George just said. He walked in and said, that's what's happening. So we were like, this is wrong, this can't be. But when we wrote to the collaborator with whom we were doing this, it turns out these worms had actually been fed on a culture of monkey cells. So it all made sense. We removed it, and we went on ahead with the project. So that was nice. This is an example where Eric Schwartz was doing this in Caltech. His favorite example for this genome is he calls it the nematode genome from hell, because he could never get it to assemble just right. He was always seeing contaminants. Again, there's no separation of GC coverage in this because it's everywhere. It's not a great assembly, obviously. That's why it's not, or maybe it's just that the bacterial contigs have the same GCN coverage as the others. But he said it helped him see that it was right in the middle of things, and he basically just did brute force BLAST with stringent cutoffs and started. But he said until he had seen this, he didn't have any idea of how bad his nematode genome was. So it's just one of those things where it's just nice to have a visualization and thing. And this one's a sample that we got in Liverpool, so that's OK. We can go ahead with that. So what's the steps for making blob plots? How much time do I have? 5 minutes? Less? OK. So this is the workflow. It's not very complicated. There are scripts for each stage of this, and I'll walk through some steps on the GitHub, which is there. So I'd initially put this up as part of my PhD thesis work on the GitHub Sujay Kumar assemblage. But when we were writing it up for the paper, I rewrote all the tools because I wanted it to use the new BLAST, because that gives you back the taxon ID. So it would have done it in one step. I wouldn't have had to run 2 steps to first get the BLAST, then look up the tables, blah, blah, blah. So that's the two where you can use it. I would use the second one now. So what happens? You take the reads, you do a preliminary assembly. Now, at this point, you have a choice. This is I think what Chris Quince called the initial co-assembly of all the reads together. We like using CLC because it was quick, it used very little RAM, but it's expensive. It's 3,000 pounds, I think, for a single seat license. I think it's more than paid off in the amount of time it saved us over the years, but you may not have so much need for it. So I find Abyss works quite well, and I will try Ray because I didn't know Ray was as easily parallelizable, because my concern was that the reason I like Abyss is I have access to a cluster with very little memory per node, but not of one single big machine. So I really would like to use Ray for this. So I would say use unpaired reads, because then you're not letting the assembler make any assumptions about scaffolding and making mistakes. All you're trying to do is reduce the search space. So that can take anywhere, for this particular project, it took about 7 hours on 8, well, it wasn't 8 cores, it was 8 nodes. So it does take time, but let's say it takes a day to do stuff. You get contigs, you align the reads back to the contigs, you get an alignment BAM file. You can also, if you want, use the kmer coverage files that Velvet and Abyss and a few other tools give you back, so you don't have to use a BAM file. You do Megablast. Megablast is quick. It's not necessarily the most accurate, but I like it because it's quick and you sort of get a sense of what you're looking for anyway. The point of this tool is not to be exhaustive, it's to give you a quick visualization. I like BLAST Plus from 2.2.28 onwards because it gives you back the subject tax ID, which is used by my script, which basically collates everything together, calculates GC, length, coverage, all of that, walks up the taxonomy tree, gives you reports. And that tabular file you can either plot using R and ggplot2, which is what I did for the plots that I showed you, or you can use a tool called Blobsplorer, which Martin Jones, who's a postdoc in our lab, just wrote in 2 days because he was like, this is stupid. I don't want to have to sit and make plots in R. So he basically wrote it. I think it's a very cool tool. It's very easy. You just download an HTML file and load up the contigs in it. But you need, it can't do very many contigs. So up to about 10,000 contigs, it's happy. Or if you don't mind sampling, you can load 100,000 contigs and work with only 1/10 of them. So it can do that. That's cool. Here are some other insights. I found BWA builds the index a little faster than Bowtie, but they all work. It's fine, whatever works for you, whatever you're comfortable with. Sorry, I've already said this. And apologies, there's yet another tabular format at that stage. But so here's the last part of my talk. In keeping with the Beatles theme, I need some help from you. Because I told you at the start I was working on flatworm genomes now, and that's the lab I'm in now. And I'm seeing an interesting blob plot that I'm hoping somebody over here will have seen before or will have suggestions for why that might be happening. So why are we studying flatworms? Well, they're about, these are planarian flatworms. They're freshwater. You turn over a rock in a stream, you'll see one. This is about 0.5 centimeter to 1 centimeter long. You cut off any part of it, and this is from a paper in Nature by somebody else. And the lines are telling you that depending on where you cut it off, how long does it take for the full head with eyes and a brain and everything to grow back. So within a week after amputation, even if you cut off a tiny piece of the tail, you're going to get back a full worm with a head. So they're little champions of regeneration. I mean, other animals will regenerate a limb here or something, but flatworms are really cool planarians because they can regenerate. So there's lots of them, and they have different regenerative abilities. So some have high, some have limited, some have, these ones have high again. I'm working on the 1.5 gigabase genome of this one over here. It's Gerardia tigrina. Sorry, you can't see this. This was a picture that I took from a slide somebody else had made in our lab. And so I'm trying to assemble the genome because the goal is eventually that we want to compare these genomes to see what are the genomic correlates of regenerative ability. So are there extra pathways? Are there different regulatory regions? Is it the same regulatory region used every time you want to generate a head? Things like that. So that should be very exciting. When I make the blob plot, this is what I see. So there's no, I mean, yes, you see, Diptera and Coleoptera, and which are insects and flies and butterflies and things, and you see a little bit of primate. But that's, there's no consistent blob. It's just little flecks, which I just assume that those are random hits because nothing closer was found, and I'm using a relatively lenient one e-value cutoff. My concern is these two blobs. So the fact that there are two of them, and they both say triclads being the order that we are interested in, the flatworms. One is almost exactly double the coverage of the other. You can't see that over here, but it's basically 40 and 80. That's the read coverage over there. So basically what we're saying is we have something that's in, I don't know if it's half the genome or what. Some part of the genome is in 2 copies, some part is in 1 copy. So our first thought was, well, maybe it's like a triploid. It shouldn't be. It's a sexual diploid. That picture was made 3 days ago by Hanae, who's in our lab, and did this fabulous little karyotypic experiment. I'm a keyboard monkey. I don't even know how you begin to do something like that, but the picture was amazingly cool, where you can see the diploidy, and it's diploid, it's 2N, there's no third chromosome hanging around. Kmer Genie is a tool that lets you plot using different Kmer values. I'm just showing you the picture for Kmer 21. It also shows these 2 peaks. If I use jellyfish, I still see, not in the individual libraries, but when I see the final picture, I still see 2 peaks. So the 2 peaks are real. They're there in the read coverage. They're there in the Kmer coverage. I'm just trying to figure out if anybody has seen anything like it. Sorry. That's it? Will it cause that? I mean, I thought, I wasn't sure. So, but the fact that it's almost half and half is just coincidence. Okay. Okay. Well, if you've seen this before and that's what, then I'm happy to go ahead with that, because that was what was stumping me. I was sure there was something more complicated, but I'm happy if that's all it is. Okay. Well, okay, I'll go back and, so maybe what I can do now is look at the bits where there's double coverage and see if there's an even number of SNPs or something. I haven't figured out a way to do that on a large scale. So that might be, yeah, okay, thanks. So in summary, there's really only one point. We make blob plots for pretty much every genome project. If you find it useful, please do. If you find faster options, please let me know. I just think it's incredibly useful to get a visual sense of what's in your data. It's really a QC tool. It's really not. It's like Fast QC if you've ever used Fast QC. It gives you a visual, lets you know what you can do with it. And those are the people I'd like to thank a lot, Blackster Lab especially, but also Abu Bakr Lab, because I'm having so much fun there with the regenerating flatworms. They're very cool. And funding with the gene pool, now Edinburgh Genomics, did all the sequencing. I'm funded by BVSRC right now, and some of the projects were with NERC, and thank you very much.
All right. There's a monkey in my worm. That's a classic already.
I hope Georges is watching this because.
And for anyone that missed that, Neil said heterozygosity was the reason, and I guess that.
I'll go back and check that.
Makes,
absolutely.
That makes sense. Okay, I'm actually going to skip questions if you don't mind, because we'll just try and keep us on time because people have got to get trains home.
That's
so we're going to go on to the next speaker.
I'll take questions afterwards.
If you look at Twitter, he looks a lot like Sid James, but in real life he looks quite different, really. Like Brian Blessed. Okay. So next up is Mike Cox from Imperial College, who's going to bring the discussion back to 16S bioinformatics. Just get your presentation up. Okay.
Thank you.
Yes, click up.
Oops. Thank you very much. It's quite nice to be back in Liverpool, actually, because so my dad's side of the family is from Liverpool, and I met my nan yesterday. And when she found out I was presenting in the Foresight Centre, she said, "Oh, when that used to be a hospital, your great-grandmother died there." So I'm hoping to emulate her performance today. I'm going to be talking about copy number correction in 16S RNA sequencing. This, I'm starting with acknowledgements because this was a master's student project with Daniel Homeler, who was doing the bioinformatics masters that's run at Imperial. He's now a student with Elaine Holmes on integrating, looking at integrating metabolic and clinical data. I co-supervised him with Geraint Barton, who's part of the bioinformatics support service there, and Sarah Butcher runs part of that course. And then also John Pinney was involved. He's a theoretical systems biologist. Daniel Money has been really helpful. He's out in Kansas, and we used one of his tools, Duplify. And then I am supervised by Miriam Moffat and Bill Cookson in the National Heart and Lung Institute. And they're funded by a Wellcome Trust Senior Investigator Award, and that's what pays me. And then finally, thank you to everyone here for invitations and funding. So disclosures, I'm a microbial ecologist. I'm not a bioinformatician, and this work is not quite finished yet. So there are a couple of questions in microbial ecology. Who's there, and what are they doing, and why? So we use 16S RNA sequencing to get at one of these somewhat. There are lots of other ways to do it as well. So we start off with collecting samples, extracting DNA, PCR amplifying, sequencing, and then trying to work out what it means. There are lots of problems at all these steps, and it's crucial to control all these very well. One of the outputs we get, one of the key outputs, is an OTU table. So you have bacteria down the side, or your OTUs, and then you have samples across the top. Most of these tables are 0, because most of the time you don't find your organism very often. And then usually there's a stacked bar plot as well. So it's the proportions of the different organisms that are the output that you look at. Just one point, because I get annoyed. This is not metagenomics. It's definitely not metagenomics. because otherwise sequencing a single 16S gene would be genomics, and some people will get very annoyed. So the important thing is that if you have one read coming out of your sequencing, that represents one genome extracted from one organism in your sample. Of course, that's not true. So over 80% of organisms with sequenced genomes have more than one copy of the 16S gene, and 25% have more than 4. So in fact, this alters the picture of your proportions quite significantly. The nice thing for me as an ecologist is that copy number also has real-world meaning. So for example, if you delete copies from the multiple copies in E. coli or B sub, you have impacts on growth rates. In communities, succession, which organisms appear first and which ones come up second, is related to the copy number. So if you've got lots of copies, you tend to be a big boom and bust kind of bug, whereas the smaller copy numbers come in later. Responsiveness to nutrient fluctuations and average copy numbers in different kinds of nutrient environments. So if you're correlating, which you always are with 16S, with some environmental measurement, then having the wrong copy number might influence your ability to detect associations. So it's something that I think might be worth taking into account, but there aren't many tools out there to do it, so that's where this came about. There's a nice publication from Steve Kemble and Jessica Green, where they have a look at this problem as well, and they show that there's phylogenetic signal there. So the copy number has some phylogenetic signal, and it's quite significant. So higher copy numbers there are represented by the bigger black bars around the tree. So if you have a phylogenetic signal, that means it's possible to correct based on the relationship of your 16S genes to each other. The problem has been known about for a long time, but we've always sort of ignored it in microbial ecology, I think. And one of the reasons was that there weren't enough genome sequences, but we've got quite a few more now. And also, we've done a lot more sequencing of human-associated bacteria recently, both 16S and genomes. So in that kind of environment, it's probably going to perform quite well because you've got lots of references. So the way Daniel decided to go about it was to use ancestral state reconstruction. So we have a phylogenetic tree with some copy numbers that are known and some that are unknown from genome references. You then reconstruct the ancestral state using some model. This is the bit I'm really good at. And you have phylogenetic distance to the known common ancestor, and from there you can work out what your copy number is most likely to be. He tested a whole bunch of models, but there were 3 that we came down to. The birth-death innovation model, this was implemented using Duplify, so that was Daniel Money's contribution. Brownian motion with Pagel's kappa, not got a clue what that is. And closest neighbor, which is nice, I like this one, because it's just the one next door on the tree. So we then set about testing the models. Copy numbers are available for a large number of organisms at RRNDB, which is a quite useful site if you're just interested in one particular organism and how many copies it might have. It's recorded there. It's a little bit tricky to access the data, so instead Daniel just counted 16S annotated genes from the genomes available at the time. And we used leave one out testing to see how well each model performed at predicting the copy numbers. So these were the results. Across the bottom, we have the predicted copy number divided by the real copy number. So if a model gets it right, it should be 1. You can see the common practice, which is not correct at all, in the grey bars. So most of the time it's wrong. And then the best performing model was the birth-death innovation model. So that was most of the time correct. So we're trying to put this together into a little useful script, which we're calling philonorm, because nobody has claimed philo and that combination of 4 letters afterwards yet, which basically we take the model and copy numbers from genomes, and we're hoping to keep that updated with new genomes. Predict copy numbers for species in the Silver reference database, and then users will upload 16S sequence data. We pick OTUs and compare that against the Silver reference, so we have them both in the same reference tree, and then normalize, providing back normalized and the original unnormalized data. If we try and do this with one of our data sets, we get quite dominated samples because we look at cystic fibrosis and bronchiectasis a lot. So some of the common organisms actually have quite high copy numbers, but you modify quite substantially the proportions in any real data set. And what tends to happen is that your dominant organism isn't as dominant as you might think, and some of the organisms that seem incredibly rare are actually more abundant. And this has potential clinical implications, because everybody always focuses on the most dominant organism. But if you correct for copy number, these can switch, so you get a different dominant organism, or they're actually a smaller proportion of the whole community. So, my slightly lightning talk, there are a few things that remain to be done. One thing we want to do is to benchmark the models, because there are a couple of other methods out there for doing this, and to see whether we've got the right one and to tweak that model. The Silver database updates fairly regularly and gets a lot bigger, so the reference tree that we're using gets more comprehensive. So we need to be able to keep track of that and keep that up to date. The bit where users upload all their reads is kind of rubbish, and the connection there is by names of species in the Silver database. What we want to do is actually use pplacer to put reference sequences onto our reference tree from each OTU. So then the user will be uploading a lot less stuff, and it will also be agnostic of however they've decided to pick their OTUs. One of the nice additional things is that if you have a 16S biomass quantitative PCR on the same sample you have 16S sequencing data on, you can also then use the predicted copy numbers to modify that value, so to correct that. So if you have high biomass from a Pseudomonas-dominated sample, which has multiple copies, versus high biomass from one which only has one copy, in fact, you should be treating those as one is 5 times bigger than the other. So we're hoping to report back this scaling factor for each sample as well, based on 16S data. And then it's going to be a little, it's going to be a web server for stupid biologists to use, like me, and then scripts available too. There are a couple of other solutions out there at the moment. PyCrust from Curtis Huttenhauer's group, which uses 16S data to predict metagenome function. At the beginning of that, they have a method for correcting copy number, because they have to know the right proportions in order to get the right functional proportions. That's quite functional. The Kemble paper I mentioned also had some scripts in their supplementary data, but we haven't been able to get those to work yet. But we want to be able to do that so we can benchmark our own models. But in case you want to use something straight away and ours doesn't work yet, then you can go and give it a go with your own data. And that's it.
Thanks, Mike, keeping us on our toes. So I've got a question. So how important is the copy number correction relative to the biases that you're going to see when you do DNA extractions?
Yeah, so so there's biases in every damn step of the whole 16S pipeline. So I kind of see this as just one of the things to be aware of and to incorporate into your pipeline if you can. We're just using it at the moment as an experimental tool to see what impact it has on the conclusions we might draw from 16S data, rather than something we're definitely going to do every time we do something.
Yeah, definitely sounds relevant. Mark?
Are there any organisms that have multiple sequences, different sequenced 16Ss in their genomes?
Yes.
And do you correct for that?
No.
And how many? I mean, how big a problem is that?
I think it's a smallish number. There's some evidence of horizontal gene transfer between 16Ss. It's more of a problem the more copies they have. The ones with more copies tend to have more variation, and it's a smaller proportion that have the really extreme copy numbers. So that's another problem for somebody else.
So you can get, because everyone uses like 95% sort of similar identity.
97.
97.
Yeah.
You can have an organism that's got two 16S sequences that are
more divergent than 97%.
Yeah, okay.
It is possible. I've not seen it in any of our stuff that we've done that I know of.
Okay, so again, in the interest of timekeeping, I think we'll stop there, but thank you very much, Mike. That's great. Okay, so just get the right presentation up. So our final presentation for the web stream, we have one presentation afterwards, but the last one on the web stream is Rebecca Gladstone, who's recently started at the Wellcome Trust Sanger Institute, just finished her PhD at the University of Southampton, and working as a bioinformatician. And so I won't say any more. You introduce yourself. Thank you.
Okay, thank you. So thank you, Nick, for inviting me to talk here and giving me this slightly worrying title that I now got to carry on with. Have we got the pointer? Yeah, sorry. I'm just going to go through my personal story, which is basically, I call myself an accidental bioinformatician. I come from a biology background, and this has been my way of being an epidemiologist that's gone, okay, right, let's get into this. Let's get see what we can do with genome sequences and go from there. And it's gone upwards and slightly out of control now, but you'll see all about that in a second. Am I not working this right?
It's the chain that's rattling against.
No, sorry, this...
The clicker. Can you help the clicker, Nick? Excuse me.
These buttons? They don't work. I'll just use the...
Oh, it's just you've got an animation.
Okay. Shall I do it on the space?
No, it works. It's just animation.
It's the chain.
So sorry about that. So there are three main projects that I'm going to talk to you about, and it started off with my PhD in 2009, where we started off with a pilot study at Birmingham with Nick Lehman and Mark Palin with 20 genomes from the Streptococcus pneumoniae. We upped that in collaboration with the Sanger Institute and did a whole collection of a study, 527 isolates. And then while I was writing up my thesis, I stayed at Southampton and worked on a project that was sequencing a whole load of Haemophilus, and that was 700. And now I've just moved to the Sanger Institute to carry on in Streptococcus pneumoniae, a similar kind of project looking at vaccine impact, but we've scaled it up slightly since my last project, and I'm looking at 20,000 pneumococcal genomes. So if we look at that in terms of what I've done so far, 2009 I'd never seen a genome in my life. Now I'm going to be dealing with 20,000, so let's see how that goes. I thought that we couldn't get through this meeting without some bioinformatic bingo. I thought we ought to drop that in there and say this is why we're doing it. Price is dropping, so we can do a lot more of these sequences, obviously. So if we get into the Southampton pneumococci, it's a bacteria that causes meningitis and pneumonia. It's carried in the nasopharynx, and we were swabbing children over the introduction of a vaccine to determine whether the structure of the population in terms of serotype and genotype had been changing, and surveillance to determine perhaps what the next step is needed for vaccine control of this type of disease. So we hypothesized that there would be a change of serotype and genotype, but we went on to, oh sorry, that's when the vaccines were introduced. We went on to include whole genome sequencing in this project. So this project had been running for 3 years already. So we had our primary outcomes already set, and we wanted to basically utilize whole genome sequences to get out the MLST and things like that that we were going to look for anyway, and then see what extra data we could get. Obviously, whole genome offers a whole lot more than that, but actually it wasn't going to cost us that much more. So that's why we got into that from an epidemiological perspective. And we used a gene-by-gene approach rather than phylogenetic or large-scale genomic analysis. So if we look now at what kind of changes occurred in the population, just in terms of serotypes, we have a constant carriage rate, which is the grey line on the top left here. The red line is the non-vaccine types, which increased to completely replace the non-vaccine types for the first vaccine. The second vaccine has more serotypes included in it. You could still see that change. Both of these are significant, and PCV was actually only introduced in 13 was only introduced in this final segment here, and within 1 year we could already see a difference for those. So all of these changes involved the vaccine types and non-vaccine types. We could determine significant changes for the serotypes, individual serotypes involved, and their associated genotypes, so the MLSTs that were commonly found with those, both increases and decreases. So we wanted to look a little bit more into capsular gene sequence because we were previously using PCR, and it could only discriminate between serotypes a certain amount. And with our whole genome sequencing, we just thought, oh, well, we can have a look in there for those that we have the genetic determinants of. So we did that for a number of different serotypes, some of the most common ones, things like serogroup 6. And I've just got an example here of what we observed pretty much for all of these serotypes, but in particular, we've got 6A here. And there were 4 different alleles for the capsular defining gene that makes the difference between 6A and 6B. We had 3 different positions at which there were variation. And now a single SNP can result in the difference between a 6A and a 6B isolate. And these have immunological impact. So we were really interested that there was all this variation there, where previously it had been thought that within a serotype there wasn't any variation or very little variation. But we found that the non-synonymous variation within a serotype was actually quite common. Also, what we had to look at was that looking for some new serotypes. So the great thing about the genome data was that as things were published saying, oh, we found a new serotype, here's the genetic basis for it, we could just go back to that sequence and have a quick look and see, is that there? Yes, it is. Oh, it's not a 6A, we found a 6D, and we've got 6F as well now, which was actually only published, I think, like 4 weeks before I submitted my thesis. So it was just like a last minute thing when the publication came out. Let's have a quick look at the data again, and there it is. So that's quite interesting that we can do these kind of things. So I also looked at pneumolysin because we're interested in vaccines. Pneumolysin is a component of potential future vaccines. And there are quite a lot of known different alleles, but most of them have been found in invasive isolates, and this is a carriage data set. So not only did we look for the ones, the original sequences, which we didn't find all of because they're actually found more in IPD, we found a whole load of novel ones, and the sequences were quite diverse, really. And the bit that was really interesting, or kind of annoying actually sometimes, was that we kept finding genes, and pneumolysin is an example of this, where it was supposed to be a clear indication of what was or wasn't a pneumococcus. But we quite clearly found, and we've got P20 down the bottom here, is an example of a confirmed pneumococcus that is much more similar to a mitolysine sequence. And the opposite way around, we've got sequences that were previously from pneumococci that we found now in closely related species, so it really wasn't helping us to define on a gene-by-gene basis the classical genes, which allow us to differentiate between different types of species that are closely related. So after doing all this work, I was trying to write all this up and starting with the Homophilus work. And now Southampton, we got a MySeq. We've got Iridis, the computing cluster was already there, so it was a really great resource. But I have some peer-reviewed evidence here that says that basically makes us a genome center. So we've been trying to get set up to actually do that, because previously we hadn't really got into next generation sequencing. This was our first go at doing it at home, basically. Again, this was a gene-by-gene approach, and we're looking at characterizing strains that were from patients with chronic obstructive pulmonary disease, and these were isolates that were colonizing the lungs. So we're looking at 50 or so different genes, MLST, and trying to determine whether they're typable or non-typable. So I had to develop a kind of pipeline for the group to allow us to do this, and identify all the software, get it sorted out, and things like that. And one of the main things was even just the simplest thing, starting with your storage data. If you're not set up for these things, you've got to go about arranging your storage data. You first get a MySeq, and we were manually uploading the data from the MySeq to the server. And then we were trying to sort out what we needed and what we didn't need, because we had very limited space. So we were doing all that, and then obviously we filled the storage space almost immediately. And what do you do then? You try and get rid of all the stuff that you think you don't need. And manually delete those files. And the problem is that occasionally you might accidentally have deleted some stuff, at which point you introduce getting the data back, but then you restrict all the permissions. Unfortunately, it doesn't help you if it's you yourself that's actually deleted it. You can only protect it so far. Somebody's got to be able to manage all that data. So we arranged for extra storage space, and extra storage space, and extra storage space, continuously trying to handle this. And while I was looking up for error images, it just reminded me of one that I had continuously popping up at me at the same time whilst I was writing my thesis that I wondered if anybody else has ever seen. It's quite annoying. So the other thing I was doing was setting up the actual workflow, so finding which tools were going to be useful for us to use, and that we could all as epidemiologists get on board with and repeat our analysis in a nice way. And Josh actually came down and helped me out with getting started on these kind of things. But I read a paper recently, and I'm a bit of a hypochondriac, so I got a bit worried when I read this. But I think it's fair for these isolates when you're handling huge amounts of data. You just want to be making sure they keep it nice and simple, doing the same thing over and over again. If you're trying to do masses of loads of different things, you can kind of get lost when you've got hundreds and hundreds of different things. So this is where I'm at now on a global pneumococcal project, which is trying to understand, again, the vaccine impact on pneumococci, but we're looking at all countries in the world, particularly Gavi-eligible countries. And we've got these 20,000 genomes, and they have all sorts of, they come from all sorts of different places, but they're from before the vaccine was introduced, after the vaccine was introduced, adults, children, carriage disease. So it's really a whole snapshot of the pneumococcal world. And the idea is to kind of use this data to make a case for doing long-term surveillance for pneumococcal on a global scale. So this is the firepower that I've got to work with. And the bottom one, we have great access to. A PCS5 here is one that the pathogens group at Sanger get to use pretty much to themselves. So we can play about a bit more on that one, and we only annoy each other if we crash it. And the other things that I've got to help me deliver the GPS project is a number of automated pipelines. So there's a dedicated team that works on generating all these pipelines at Sanger so that we can do these large-scale genome projects in a really robust way. And we've got it for a number of things is really useful. The QC, assembly and annotation is automatic. The mapping on request there, and then variant calling. And then they're in development of pan-genome, which I'm quite excited about. So apparently we've all got pipelinitis, but sorry about that. So this is all from Jackie's team. These are some of the data outputs that we see from the QC pipeline, which is presented in a graphical, in a web-based system that you can look at for any sample. And it just samples a certain amount of the data to generate a whole load of different plots that are useful to us, which really helps in handling such large data rather than generating this all yourself. It's just nice and ready there for you. Again, we've got a mapping pipeline. I'm not going to take credit for any of the details or anything of this, so you can just take a look at it yourself. There's uses a number of different tools and gives out your usual BAM and a whole load of stats, which are also very useful to play around with to get a feel for how good your data is, or pick any nasty things out, which sometimes happens. This is the assembly pipeline. Again, I just want to kind of show this, that there is some structure to it. It's not my thing, but I thought you might be interested to see it. And they, at Sanger, they also have different ones for viruses and for different other organisms as well. And then finally, we've got the annotation pipeline, which uses Proca and goes through all these databases in that order. And this is all stuff that's already been done on some of the isolates that have been sequenced so far. So 1 month in, what have I actually done? It's actually going to be a month tomorrow, so it's quite a big project. I haven't done it all yet, I'm afraid. 20,000 genomes is still waiting for me to deal with. So I've had my general induction. I've even had a lab induction. I'll tell you a bit more about why that is shortly. And importantly, we've had an amazing fireworks show, which is a great place to work. You get to watch this kind of thing. And I've logged about 200 kilometers on Strava getting to and from work so far. So actually, what have I done? We have 1,000 sequences available at the moment. And I've been told off a few times already for saying only, because apparently 1,000 is quite a lot. I just think, oh, I've got to get these 20,000 done, and now 1,000 suddenly seems small. So again, I said the assemblies and the annotations are already available, and I've been kind of dealing with data organization, so trying to figure out what we have received, where it's come from, getting the metadata together in a usable format, trying to figure out what's worked well, what hasn't worked well, what needs repeating, these kind of things. And actually, we need 1,000 sequences or 1,000 genomes is quite a task in itself. So I've been going through the manual data curation. I've just run it through the MLST script that they have at the Sanger Institute, which is assembly-based. I've also ran the SRST2 as well on it, just for a little bit of a comparison between the two. And I've looked at the serotype using an in silico serotyping PCR. And this is all to help me investigate some of the teething problems that we've had. So basically, some of the problems that have been so far have been real logistical problems. So we've got 5 main sites at the moment. We're probably going to have a lot more because we're still looking for samples from certain areas of the world that haven't been well covered yet. So plate seals have caused a problem, things getting sent across, seals coming off, things getting contaminated in transit. We've also had, obviously we have to deal with things that are coming from climates that are a lot warmer. Some things, they don't have the facilities to store them on cold temperatures. It doesn't come on dry ice. Everything's just shipped at ambient temperature, and it arrives when it arrives. We started off with a grant that bought a whole load of robotic extractions, because some of the sites were extracting 3,000 or 6,000 isolates. So they wanted to make it as automated as possible. Unfortunately, after a year of trying, robotic extraction has always been completely thrown out now. There's only one site that's still trying to optimize it, but we're spending more time optimizing that and then getting kind of low yields and rubbish data than it's worth. So a lot of people have just given that up and moved over to manual, which is obviously a lot more work. And so we've had to basically resequence a whole load of isolates, because we had these kind of low DNA yields coming in, and some projects you can kind of go, right, okay, we'll see what we can get away with, might get good data out of it anyway. But that hasn't always been the case. So we've had isolates that have come And we've had a second go at sequencing them. We've had isolates that have been sent multiple times. And actually, that's what I've spent an awful lot of my time doing now, is working out what's been sequenced, when it arrived, and which batch it arrived in, and trying to collect the actual data that we're going to include in our analysis. And this is only for 1,000. So it gets complicated when you're dealing with people that have their own protocols and sending their own isolates in, and then trying to work from there. But we think that we've got it all sorted out now, and everything's coming in nice, and the data's looking great. But the one thing that Steve Bentley said to me that we need to be really careful for looking out for is checking all the metadata, because people potentially can be sneaking in their own projects under the global thing and we can spot some of those. You see any patterns coming in the metadata, and you can have a look for that.
You know, he only says that because he used to do that.
Probably. So I just wanted to mention this, the MLST from genomes. Okay, yeah, it doesn't tell you everything, but I think it's still useful these days, and people still want to know what the ST is, even if they are doing large-scale genomic analysis. But my sort of worry is that the usefulness of the databases are really dependent on what people are submitting to them in real time now, and we're getting all this data from whole genome sequences, and I've had a couple of contacts with the databases trying to get these submitted, and conversations have taken like a few months to decide, well, maybe we would like FastQ or VCF, and I have submitted a few. And I haven't actually had any allocated results yet back from things that I've submitted for whole genome sequences. And I just wanted to point out, I guess, that we're going to lose the value of these schemes unless we find out a way of being able to submit these in large numbers to the databases themselves. So this is just the beginning of this project. With 19,000 left to sequence, I've got 4 years, hopefully, to sort myself out. And the analysis is really to come in the future. So yeah, watch this space, and we hope to be recreating some of the analysis that Nick Croucher has been doing on looking at vaccine impact on pneumococci and the Massachusetts data. And my main sort of quick tips really that have nothing to do with bioinformatics at all is that actually, when you're dealing with projects this large, you really have to get ready to project manage in a big way and deal with all the different sites. Spend a lot of time tracking samples and data, and the really simple things you can do to make your life easier is obviously cool things, informative names for your files and folders, and to set up your hierarchy and databases in advance. Don't try to remember everything, which was my old trick, and literally I have to copy and paste everything that I've done as I do it into books at the moment, so that I keep a trace on what I've done, and make yourself disposable essentially, so that if you get hit by a bus tomorrow, somebody else can easily determine what's going on with it. So my major sort of point really is start organized. So I've got a few people to acknowledge from Southampton, my supervisors, Stuart Clark, Joe Jeffries, and Sol Faust, and then the Sanger Institute as well. Stephen Bentley is my new boss there, and the pathogen informatics team, because they're helping me to handle all these in bulk. Well, thank you.
Excellent. So, I mean, it is miraculous. Mark Palin just tweeted, it's miraculous to go from 20 genome sequences to thousands of genome sequences in such a short amount of time, and trying to cope with that is obviously a challenge for all of us. Time for one quick question. Anyone want to make a question, have a question or a comment? Okay, all right, well, I mean, this segues quite nicely into the next talk. Thank you very much, Rebecca. Thank you. Oh, yeah, no. So okay, so I must say, we're going to end the web stream now, so we don't need a mic, actually. So thank you very much for watching if you're on YouTube. See you later. We've got some very important stuff to discuss that you can't see now. No, Chris Heyman from Amazon is going to talk next, and he would have to go through various form filling to get his talk webcastable. He has said that he will see about, we'll record it anyway, so he was going to see, so, we do need it, so what am I talking about? So he's going to see if we can post that later on for people that are interested. So it segues nicely in terms of what Rebecca was talking about, because if you don't have the Sang as infrastructure for your genome sequencing, maybe you need Amazon Web Cloud, and you can build a data center probably as large as the one that Rebecca was talking about from your laptop, and hopefully that's what Chris is about to demonstrate. So without further ado, after you, Chris.
That's great, thanks, Nick. And I appreciate I'm in between you and beer, so I'll try and keep it brief. I've got a demo as well. It's quite brief, but it will demonstrate Ray running in parallel, so I'm sure pleased to hear about that. So what I really want to talk about, so effectively I'm a solution architect. I'm focused on UK public sector and primarily academia as well. What I really want to talk about is talking about some of the building blocks and services you can use within the Amazon cloud to hopefully improve the speed and agility of which you deliver scientific results. So that's the goal. So what I'm primarily going to focus on is the compute storage bit of the Amazon cloud, only a very small subset of what you can buy from us, but I think it's the most important in regards to this particular presentation. I'm just going to briefly mention public data sets as well. I'll come on to those, Amazon Genome being one of them. And then I'm going to talk about some managed services that we provide as well that may or may not make the life a bit easier, I don't know. And also some more traditional high-performance computing tools as well. So this is kind of what we deliver from within Amazon as part of our Amazon cloud. We have a product offering in pretty much every one of these boxes, but